Mm.
Yes, on behalf of the organizing committee, I am excited to open the virtual day two of the conference, which is the 16th World Conference on Midwifery, Nursing, Neuroscience, and Brain Health.
Mm. Mm.
Yes, please let me welcome our first oral speaker, Grigory Tissoni from National Institute of Mental Health and Czech Republic, to deliver his presentation on neuroactive steroid, pregnenolone, glutamate, anti-epileptic and neuroprotective and anti-inflammatory effect in perinatal focal cerebral ischemic model. Yes, Gregory, please start your presentation.
Mm. Mm. Hello, are we starting the presentation or what's happening?
Yes, sorry for the interruption. Maybe the Gabriel is not available right now. Next, moving forward to the next presentation. Now, I would like to invite Bilal Amin from University of Cambridge, from United Kingdom, to present his presentation on mitochondrial DNA in glioblastoma, a systematic review.
Again, is this presentation going to take place or what are we waiting for?
Hello, can everyone hear me?
Yes, yes, Bilal.
And can you see my
Yes, it's okay.
screen?
Yes, I see the screen.
Okay, hi everyone. Sorry for the delay. My name is Dr. Bilal. I am a first year resident doctor in the UK, and recently graduated from University of Cambridge, where I did a systematic review on mitochondrial DNA within the context of glioblastoma. Apologies, I thought my presentation was at 11 a.m. So I was just getting some files ready. Okay. So just some contextual information. Glioblastoma is a highly aggressive primary cancer affecting astrocytes. In patients present with symptoms of raised intracranial pressure, meaning headache, nausea, seizures, and oftentimes we'd see focal neurology on examination. Um, it's a highly infiltrative disease, which means that it's usually not or patients with glioblastoma are usually not candidates for complete surgical resection, and its heterogeneity limits what chemotherapy can do. So generally the gold standard for treating glioblastoma is a combination of surgical resection with adjuvant radiochemotherapy. But despite this, the disease remains quite rapidly and uniformly fatal, with an average on treatment survival time of only 15 months. Um, and uh, on the side of that, variations in mitochondrial DNA have been reported in multiple cancers, and that includes glioblastoma. Sorry, I haven't got my video on. Is it on now?
Yes, yes,
Yep.
perfect.
Okay. And that includes glioblastoma. Um, Often detecting these variations in the mitochondrial DNA is complicated by something called heteroplasmy, which is the coexistence of multiple um, mitochondrial DNA molecules within the same cell. Of course, because we have thousands or hundreds to thousands of mitochondria within a single astrocyte, and then each mitochondria also contains 2 to 10 copies of its DNA. So the objective of this systematic review was to simply synthesize all of the data relating to mitochondrial DNA within the context of glioblastoma. So the way we went about this was first developing a robust keyword search with our librarians. I've put sort of the details of that in a table format on screen. And then we then searched for databases, Medline, Embase, Web of Science, and Scopus, up until October 2023, which is when we started the project. We removed duplicates and then applied our inclusion-exclusion criteria. So we were looking for primary studies which were in English and had full text available to us. The conflicts were resolved in a Zoom meeting, and we used the Joanna-Briggs tools to identify risk of bias following manual data extraction. So, what were the results of the search? The search returned a total of almost 10,000 records, but almost half of these were duplicates. So, after deduplication, we were left with just over 5,000. And then, upon application of our criteria, inclusion and exclusion criteria, we were left with 35 studies to be included in the study, or in our paper. We then went on to thematically categorize these studies. So obviously there's some overlap between between these categories. So some studies featured content of multiple categories. But broadly speaking, the majority of papers reported on mitochondrial DNA copy number and mutations, with a few papers reporting on gene expression and epigenetic changes. Also, to further categorize these papers, only 4 made use of mouse tissue, with the remaining 31 studies using human tissue or human cell lines. And a Prisma diagram is also displayed on screen at the moment. So the first theme of our study was mitochondrial DNA gene expression. We found that the literature suggests that compared to tumor adjacent histologically confirmed healthy brain tissue, glioblastoma displays reduced expression of multiple proteins, but Keely, these are, or mitochondrially encoded proteins. Keely, these are all involved in the electron transport chain, which links, which sort of just highlights the link between the prominent Warburg effect in glioblastoma and these mitochondrial changes. So the Warburg effect, just to quickly summarize, is a metabolic reprogramming from oxidative phosphorylation to glycolysis. And obviously, if you've got mitochondrial involvement or changes involved in a cancer process, that's going to impact that's going to impact OXPHOS and the proteins that are being encoded by the mitochondrial DNA. And we found one study that also demonstrated a gradual decline in OXPHOS complexes with a rise in tumor grade. The next theme of our study was mitochondrial DNA copy number. So this was interesting because there's quite a lot of conflicting data in the literature. 3 studies suggested an up to 25-fold increase in mitochondrial DNA copy number. I have them listed in table format here. 2 studies reported no change in mitochondrial DNA copy number in glioblastoma cells versus normal healthy tissue. And 5 studies reported a decrease in copy number. And interestingly, these sort of dual findings of both increase and decrease were also observed in nuclear encoded transcription factors, with some, such as TFAM, with some studies suggesting reporting sort of greater levels of TFAM and some studies reporting lower levels of TFAM. We think that the sort of both increased and decreased copy number make sort of logical sense, because you could argue that a higher copy number is a reflection of the anabolic state of the cancer, whereas you could also argue that a lower copy number is a reflection of the Warburg effect and the de-differentiation, which is in line with sort of the cancer stem cell theory. The third theme was epigenetic control of mitochondrial DNA. So there weren't very many studies commenting on this. One lab performed 2 nice experiments that demonstrated up to 25 sites of differential methylation. And they also displayed that the differential methylation impacted copy number. They went on to perform another experiment in which they used demethylating agents to establish somewhat of a more causal relationship. And they also identified that ND5, again, another mitochondrial gene coding for a part of the electron transport chain, was the most methylated gene. But overall, these varying levels of methylation didn't correlate to any changes in gene expression. So the significance of these methylation levels is yet to be determined. The fourth uh, theme of the research was mutations, and this was the other bigger theme amongst the papers. So just briefly regarding mutations, in mitochondrial DNA, mitochondrial DNA experiences a 10 to 100-fold greater mutation rate compared to nuclear DNA. And that's because of a lack of protective histones and the higher rate of replication and a lack of also efficient repair systems. And again, like I mentioned at the start, the analysis of what is significant or what is a significant mutation within mitochondrial DNA is complicated by heteroplasmy. But overall, the literature suggests a low burden of somatic mutations, but a significant burden of germline mutations, with a ratio of germline to somatic mutations being 21 to 1. Of course, there were dozens to hundreds of mutations listed across all of the studies, but I've just highlighted a couple of the more significant ones. Um, for the first one I've got here is the T14798C mutation, which is a substitution that we find in up to a third of glioblastoma cases. But interestingly, actually, with this mutation, it's positively correlated with glioblastoma cases in the UK, but we don't actually see that correlation within US cohorts. But nonetheless, it's present in around a third of GBM cases. And mutations to the ND4 gene were the most frequently detected. Okay, and now sort of the more interesting side of things. So what does all of this mean, really? Is there any real world use to actually looking into this relationship between mitochondrial DNA and glioblastoma? One of the questions we wanted to ask is whether or not mitochondrial DNA can be used as a biomarker in glioblastoma in clinical contexts. And of course, a marker or a good biomarker would be one that's accurate and non-invasive. We found a study that reported that mitochondrial DNA is a more sensitive indicator of the presence and burden of a glioblastoma tumor compared to nuclear DNA in not only plasma, but CSF and urine. So that presence in sort of multiple bodily fluids means that it is easily sort of acquirable. And if we're able to detect it quite nicely, we now need to know that it has some real meaning. What's the purpose of detecting it? So multiple studies actually reported on prognostic value or the prognostic value of mitochondrial DNA. We see that when looking at copy number, survival time seems to vary, with one study reporting an average of 10 months survival time in glioblastoma with a low copy number versus 18 months in high copy number. But then this may be an age-dependent relationship. So another study found that was only present in younger cohorts, and that the opposite was true for older cohorts. Again, there's not many papers on this to sort of further clarify. We can also see that with the T14798C mutation that I mentioned earlier, carriers of this tend to have a worse survival time with an average of 373 days versus 415. And also looking at the expression of certain genes, TFAM and POLG were noted to be or have increased expression in cases surviving longer than 24 months versus cases less than 12 months. And the final theme was or question was to look into how these mitochondrial DNA changes related to therapies. So one question we wanted to think about was, could mitochondrial DNA be used to guide chemotherapy drug choice? We found a few studies looking at the effect of some sort of change in the mitochondrial DNA on chemotherapeutic agents. So these studies tended to use IC50, the concentration of a drug required to inhibit a biochemical function by a half. And one study found that mitochondrial DNA depletion reduced sensitivity to temozolomide, but also increased sensitivity to cisplatin. We also wanted to think about a potential for any new drug targets. So the T14798C mutation is one that affects the cytochrome C oxidase subunit 3 protein, and it seemed to convey an increased sensitivity to clomipramine. And finally, regarding mitochondrial changes in relation to radiotherapy, there was again sort of this overall unclear picture due to contradicting findings, with one study reporting that radio-resistant tumors are associated with a greater mitochondrial DNA copy number, whereas another found that these same radio-resistant tumors were associated with an upregulation of a certain protein that was actually a DNA, mitochondrial DNA suppressant, leading to lower copy number. So where does that leave us? It seems to really be sort of quite a mixed bag of findings out there. I think it's clear that there is definitely a role for mitochondrial DNA alterations within the biology of glioblastoma, and there seems to be some sort of link between how the disease progresses and the therapeutic response of it. So there is sort of promising data for mitochondrial DNA as a biomarker for diagnosis and prognostication purposes, and perhaps even guiding treatment. But the findings remain sort of overall findings remain very unclear. More research is definitely required in the field. I think a good starting place for that further research would be perhaps a meta-analysis looking at mitochondrial DNA copy number in glioblastoma, but also other directions that could be taken down include further looking into this relationship with age and geography. As I mentioned earlier, there seems to be opposite findings based on sort of older and younger cohorts and US and UK cohorts, but also the sort of that T14798C mutation seems to be quite significant if it's present in up to a third of glioblastoma cases. So maybe looking into COX-3 inhibitors like antimycin and seeing if there's a different impact of that drug in patients with the mutation present versus those without. Those are my references, and happy to take any questions.
Yes, thank you, Bilal. Thank you for your such an informative presentation. If you have any questions to Bilal, please post them in chat, or you can directly unmute and ask him directly to answer your queries. Yes, I hope there were no questions for Bilal Amin. And once again, thank you for your wonderful presentation, Bilal Amin.
Thank you for having me.
Moving forward to the next presentation, I would like to invite Pankuri Sharma from Manav Rachna International Institute of Research and Studies from India to present her talk on eyeballing the brain with neuroinformatics.
Good morning. Am I audible?
Yes, perfect.
Okay. Is my presentation visible? Um, Ma'am, is my presentation visible? My poster is visible?
Yes, it is.
Okay. So I can just zoom in a bit over here and then probably move to the next part once I'm through with the introduction. Okay. Good morning, everyone. My name is Pankari, and I'm presenting a poster titled Eyeballing the Brain with Neuroinformatics. During this talk, I will be talking about the recent tools and techniques and advancements in the field of uh, neuroimaging with the help of neuroinformatics. Now, neuroinformatics is basically an interdisciplinary science which has been created by merging neuroscience and computer science. And this is created basically because uh, when we talk about neuroimaging, there are MRI scans, there are fMRI scans, there's a lot of microscopy going on when you have to visualize the neuron. So a very vast amount of data is created, huge data is created, which is very hard to understand and make sense manually. So to overcome that barrier, we use AI and machine learning to make sense of that data and to extract meaningful information. So with keeping that in mind, neuroinformatics is being used and the main focus of my research is Huntington's disease. And during that work for my PhD, I have come across troubles and problems wherein I had to choose and I required tools. Uh, so, with that mindset, I created this small study where I assessed certain number of tools which are available. Uh, we did a comparative analysis for these tools to give clarity on which tools can you use, what tools are available, and which tools can you use as per your requirement. Uh, so as I talk about the objective, it is to assess the AI integrated neuroinformatics tools that help in visualizing and analyzing large complex brain data. So for this, I did a certain text mining using Python with search words. I took somewhere around 130 search words. And on the basis of that, we selected the neuroinformatic tools and basically the criteria was their relevance to neuroimaging, their support for and integration, how AI and machine learning workflows were integrated within these tools, whether they are open source or not, uh, can they work with large data sets, again, how easy were they to deploy and were they beginner friendly. So these parameters were kept in mind while selecting the tools. And then each tool was compared for its unique features, strengths, limitations. And the summary table is available over here. Uh, I will talk about each tool that I selected for the study. The first one is G-tree. G-tree stands for Global Tree Reconstruction System. Now, this tool, the main focus and purpose of this tool is to help with neural tracing. Now, if I see if I show the image over here, uh, so yeah, so when you have stacks of data like this, then you can actually use these tools. Like you can use G-tree, and you can actually trace the neuron exactly. And in the second image over here, you can see that they have traced 5 neurons successfully for visualization. So basically, G-tree helps with 3D reconstructions. The data it supports could be electron microscopy, light microscopy, connectome data, and the scalability goes from medium to large. Uh, it has a graphic user interface and can be worked on different platforms. Uh, can I just, yeah, okay. So it has high quality 3D reconstructions. Now, the only problem with this is that a certain amount of coding is required. You need certain manual input is required. It is not completely automated. Uh, and But however, the key strengths remain that it can make retracing or reconstruction of neurons in 3 dimensional very extremely precise. So when you want to view neurons as such, then G3 would be your tool to go. Moving on to Newprint, this takes in connectomic data. Now, why connectome data? You would have a question that if you're talking about neuronal imaging or neuroimaging, then why are we talking about connectomes? So basically, connectomes are it's basically the information wherein how one neuron is connected to the other and how the neural network as a whole is being formed and working together. So when you want to understand that thing, then you will work with connectomic storage and connectomics data and those queries. So Newprint helps you build these connectomes. It can take in large amount of large scale connectome data, probably from electron microscopy. And it has an interactive web interface which is easy to use. Uh, like Unlike G-tree, G-tree is for reconstruction. This helps in managing and understanding large neural networks, not just one neuron, two neurons, but as a whole network, you can understand that with Newprint. Again, it is scalable for very large connectomic data. Uh, Machine learning and API for custom pipelines, web user interface, as I told. And it is very easily integrated with other web queries like CatMade. So CatMate is I've given the full forms also here in the abbreviations I've explained over here. Collaborative annotation toolkit for maximum amount of image data. So basically, it's a annotation tool wherein you can collaborate large amount of data and you can annotate it and make sense of it. So next is the NeuroDesk. Uh, this talks about containerized neuroimaging tools. Uh, this is in itself, in its own sense, this is not a single tool. You can call it a platform, or probably, the word says tool suite. So I will show the workflow over here. Now it is a community-driven yeah. So this is a community-driven suite. Uh, Many scientists, developers, researchers, they can contribute to this. These are basically the containers, neuro containers. Now each container has its own tool. It has its own settings. It has its own libraries, and you can access whichever and whatever you require. So over here you can see, our studio is here. MNE is here, free surfer. These are just examples, but it can hold like Neurodesk as itself can hold n number of containers, and at one single point in time, you can use multiple containers if you want to. If you have certain data and you want to run a query with different analysis, then you can do that very easily. The major advantage is that multiple pipelines can run together with different parameters. It cuts down on your startup time, that you have to, it standardizes the process and makes things much more easier work-wise, and it gives you two interface. The computing environments are given over here. It can either work on your Linux, Windows, and they have a normal like the interfaces as such. Otherwise, you can always go for command line if you have interest in coding, and if you understand coding or you have any knowledge about it, then you can definitely use the coding for that as well. Uh, so the major disadvantage for Neurodesks is that it is very very large. Somebody who does not have, like, it is not beginner friendly, because you have to select the tool, and on the basis of that tool you have to select the right container and then download that much data. It will run on your local disk. Uh, However, the most important advantage still being that it's easily accessed. There are multiple tools in one software with the same versions, with the updated versions running in real time, and you don't have to worry about the version changes or all of those things, and your results are easily reproducible. So that is Neurodesk. Next is Neuroglancer. Neuroglancer is more like Napari, but Napari is basically, again, just a viewer for if you have any neural data and you want to visualize how it looks like. So you can go for Napari, but Napari is more of a local kind of thing. It's going to work, but it has a limitation for data. If you have a huge amount of data, Napari is not going to work for you. Whereas if you go for Neuroglancer, it works on cloud storage. Data is not a problem. It is very, very scalable. It works with Google Cloud, and backend. And it is, again, it's browser based, handles uh, TB, data sets. So uh, the problem with Napari, this solves. But the only problem with this is that it has no analysis. It does not give you the option for doing any kind of analysis. It is just a viewer. So if you just want to view your data, visualize it, make sense of it, then Neuroglancer is good for you. But if you want to do any kind of analysis or any downstream processing with the data that you have, then you can choose from any other of these mentioned tools. Moving on to BrainGlobe. BrainGlobe is a Python-based image analysis and visualization. So it works with large data, brain atlases, neuronal morphologies, and it can again work with medium to large size data. It has a very strong machine learning integration since it works on the Python ecosystem and Python API. So here I was talking about Napari, right? The visualization tool. So it is integrated with the Napari plugins. And again, it's cross-platform. Uh, but it requires Python coding skills. Since it's working in the Python ecosystem, it requires Python coding skills and would not be that beginner friendly. So all in all, if I talk about these tools and which one would I select for my study. So what I understood after going through the advantages, and all the things that all these tools can do, uh, I understood one thing, that neuroimaging in itself, it's a very huge thing, very vast domain to study. And if you want to just visualize your data, like just visualize and see how the neurons are looking, how the connectome is looking, then probably, you can go for a neuroglancer, or you can just go for new print and you'll be done. But it mostly depends on the type of questions you're asking. What is your query? What do you want to do with that data? So if I was supposed to do an analysis, then what I would do is I would probably, go for G-tree to uh, get sense about my neurons, do a 3D reconstruction and get to know my neurons, how they look like. Then probably go for connectomics and get a wider neural network out of those. Once I have the network, then probably I would go for brain globe for the further analysis. So basically, no one single tool is going to solve the problem at the moment. So at the moment, these are the visualization tools and analysis tools when it comes to neuroimaging, and not one tool is going to solve all of your problems. So what we can do is that, as per our requirement, as per our need, we can use these tools, utilize them, and the most advantageous things about these tools are the ones that I've selected for the study. They're all open source. Probably completely or partially. Some aspects would be, obviously when you talk about cloud storage and cloud this thing, uh, uh, then obviously when it comes to large data, obviously that is going to be paid, right? So the tool in itself is open sourced. So you can use these tools depending on the type of query that you have. And in the future, probably, I think that Neurodesk, the one that I spoke about over here, with all the tools that are integrated into one, I think in the future, Neurodesk could be a really, really vital tool for us to understand how the neurons are working, how they are connecting, how the synapses are working. So all of those things, one suite can do for you. So that is one advantage of Neurodesk, the reproducibility, the multi-tool concept, the multifunction concept that it holds. And in the future, uh, if we could have something integrated into one single tool. Now, when we talk about AI, when we talk about machine learning, even these fields are, if you talk about them, in the future, that would be wrong. These are present. It's happening. AI is happening. ML is happening. So these need to be more seamlessly integrated into these tools, or probably other tools. But the integration needs to be seamless so that researchers who are, beginners, who don't have the specialized training, they can actually make use of it. When you talk about coding systems, and we talk about the Indian scenario, young researchers who are into this field, they are not very susceptible to these things. They don't understand these things very easily. They have to be trained. And then again, a lot of resources go into that. So my take out of this is that in the future, these technologies will boom. Most definitely, they will boom as AI and machine learning are growing, and the way they are being integrated into systems. This definitely will be not a problem of tomorrow. I think we are growing, uh, and I think we are expanding. So this was my talk. Thank you so much for attending my talk, and I would be open to take up any questions and queries related to this. Thank you.
Yes, thank you. Thank you so much, Pankuri Sharma, for your wonderful presentation. If anyone have questions to Pankuri Sharma, please unmute and ask her directly to answer your queries.
I would have one question, if possible.
Yeah, yes, sir. Please tell me, sir.
Uh, for instance, I study, is not my case, but I give an example. I study a subject 8 hours using an fMRI. Maybe I can use also EEG. I read some parts of his brain or her brain, and the person visually observe different photos, and then I can predict without knowing at which photo the person sees, I can predict that that person perceive a car or a tree or something like this. But the problem is that I use fMRI for this person after 7, 8 hours. If I use fMRI for another person without training, I cannot guess the photo at which the person see. So your very interesting studies with different programs and so on and so forth. But the problem is, my question is, these programs are applied for different person after a huge amount of training. But if I use for a new person without training, can I use the information that I collected from
Yeah,
different
you
persons?
can. Yes, yes, indeed, yes. You can use that information as a standard, right? You'll have something
Yeah.
to compare with. So when you have a native data with you, uh, then you have a standard or a baseline to compare it with. So that will obviously help you. You'll not be starting from scratch. So in that case, I would suggest that understand the neural network, the networks that are forming. And then and once you study the network and once you get the idea that in this case, this person is seeing something else and perceiving something else. So you have a baseline that how the network looks like, what is being activated, what is being dormant. And with that baseline, you can definitely compare it for the new person. I mean, the data will be definitely different, but for a comparative sort of a study, you can definitely do that, and you'll be at one step ahead. You'll not have to start from scratch in the second time.
Okay. Thank you. Thank you.
You're welcome. Yeah.
Yes, any more?
Shall I stop sharing?
Yes, you can.
Thank you. Thank you everyone for attending my presentation.
Yes, next I would like to invite Wilfred Angie Abia from the University of Yonet from Cameroon, Central Africa, to deliver his presentation on dietary exposure of neurotoxic mycotoxins in Cameroon, implications for brain development and neurodevelopment disorders in children. Yes, hi Wilfred. You can start your presentation.
Hello, good morning, Laura, and good morning, dear
Yes,
participants.
very good morning.
All right, thank you
Yes.
so much. I want to begin. Oh, great. So I want to begin by hugely thanking the organizers of this very important meeting for giving me the opportunity to share a bit of my work here in Cameroon with great minds across the globe, hoping that there could be further gatherings and further collaborations. I also want to say a big thank you to all the distinguished speakers that began yesterday and those that have done tremendous job early this morning. My assignment for today is to share with you about how the food that local Cameroonians eat may have an effect on development of the brain and especially for young children. So I am Wilfred Anye Abia as introduced, and I hold a bachelor's and master's degree in biochemistry with expansion into nutrition and food safety. And then I worked up specialization in toxicology, and later on have continued to assist and also serve different companies and different universities across Africa, and have had a couple of postdocs across South Africa, Austria, United States, and the United Kingdom, where I spent most of the time now, although serving as a senior lecturer of biochemistry in the University of Yaoundé 1. My whole research has been looking at food and its relationship with human health. And I have spent most of that time doing it in Cameroon, but collaborating with a lot of other great minds across the globe. So I sit today in Yaoundé in Cameroon with a population of about 29.88 million people in Cameroon. And in the capital city where I am, we are about 4.854 million of people. This is just a fair view of how our central city looks like. I hope it would be of interest to some of you to want to have a feelings at some point in your career. So my discussion would just give you an idea as to what may be present in food and how this may be connected with neuro developmental disorders in Cameroon. And I take particular interest in this because we have a society where medical system has actually been almost the direction, but I am one of those who have continuously insisted that if we know as much about food as our medical personnel know as much about drugs, we can be able to reduce health disorders by over 60-70%. So that journey has been started, and I continuously take interest in food contaminants on one hand, and on the other hand, health disorders that may come up as a result of exposure to contaminants in this food. And I am particularly interested in vulnerable groups such as children, pregnant and breastfeeding mothers, and also I take interest in looking at diseases that may actually show up for this vulnerable population. So for the last 3-4 years, I have spent a lot of time looking at food contaminants and cancer, because 10-15 years ago, cancer was just, almost not an issue. But within the last few years, we have experienced a whole lot of cancer patients, including senior academicians and researchers falling to cancer. And so that caught my attention. And of last year, I began thinking about what are the diseases that are growing up in the country and in the surrounding African areas. And neurodevelopmental disorder became one of them. So I took special interest and set up a small team for that. And we started looking at a small project, which I want to share with you how far we have gone. So I will begin to talk to you about, what our focus really is, and looking at the different work packages and how far we have been able to go. So overall, this little project is to examine the potential impact of dietary exposure to neurotoxic mycotoxins on brain development and the risks of neurodevelopmental disorders in children in Cameroon. So Cameroon is a little country in the central part of Africa, and we have 10 different regions. And I am sitting in the center region, which is the administrative capital of Cameroon with the red dot. And most of the work is anchored from Yaoundé, but spans across different agroecological zones of the country. So this little project focus on the first thing is for us to perform a lot of desktop-based research and dietary and epidemiological survey to understand exactly the mycotoxin dynamics and also the neurodevelopmental disorder situation ongoing in the country. And the next phase or package of the work is to look at dietary mycotoxins exposures, and by monitoring, looking at these chemicals, these contaminants in the food and in the blood urine samples of consumers, and try to correlate this and look at predisposing factors and advise so that policy can be put in place to cut down or keep the people far away from this harm. And the third package is to look at neurodevelopmental disorders vis-a-vis exposure to these mycotoxins, where we in turn to work with animals, experimental animals, and primary school children. And the fourth part would be packaging all of this and proposing the direction for policy and introducing a couple of capacity buildings. So the first work package aims at assessing level of knowledge, attitude, practice of pregnant women age, and also pregnant women that are also at breastfeeding stages, and how they interact with their society, including the environment and the food that they have, and what disorders do they frequently come down with. So to accomplish that, which we began in 2024, we have been able to scout the literature and have been able to publish one great paper on a scoping entitled "A Scoping Review on Mycotoxin-Induced Neurotoxicity." And we have just had the second grade review paper received and accepted with minor revision on neurodevelopmental outcomes of mycotoxin exposure and effects on brain development in infants and young children. And the third one is ongoing, which is built upon the discussion I'm going to share in this meeting, entitled Dietary Exposure to Neurotoxic Mycotoxins and its Implication for Brain Development in Children, Cameroon. So I would now share with you a summary of what we have in these 2 to 3 papers that have been produced. So the first thing is that a lot of great minds have already talked about brain development yesterday and today, I would presume. And our interest lies from prenatal right up to early childhood on the 5s. That's the spectrum we are interested in. And of course, we take special interest to understand what happened from neurogenesis through neural migration, myelination, synaptogenesis, and pruning. And of course, just to refresh and summarize some of the already great information passed out, neurodevelopmental disorder involves a set of disorders that attack or affect the brain during growing and may affect functioning. And that may not just end at the childhood stage, but may accompany this individual all through their life. And amongst these, those that are actually of high frequency occurrence, especially around here, is autism spectrum disorder and also attention deficit hyperactivity disorder. And it is of interest for us to find out how, if we go to the field, how can we know that children may be suffering from this kind of disorder? So language and speech challenges are common signs, difficulties with learning, social skills deficits, abnormal behaviors, and impaired motor skills, which of course may require us to do a bit of laboratory, but we focus a lot on the behavioral issues. And of course, basic tests like neurologic examinations can be performed to understand what is happening, and we're going into that direction. But coming down to Cameroon in particular, from Africa down to Cameroon, in Africa, tens of millions of individuals are already known to be suffering from autism spectrum disorder. And in Cameroon, we do not have that clear-cut data, statistics on this neurodevelopmental disorders so far, but reports from the Ministry of Public Health have reported that over 100,000 children may be affected. And one other thing that is actually confluencing this whole disorder is the fact that in this side of the world, Cameroon, like in Africa, most societies would think that the disorders like autism are associated with witchcraft and curses to particular families. And this turns to impede these families from seeking medical attention and rather going towards mystical healings, which many times end up not helping the situation. In terms of attention deficit hyperactivity disorder, clues that we have from a single study in Cameroon reveals that the prevalence may be about 24.4 percent, whereas in Africa the clue stands at 7.47%. So all this put together just let us know that there is a need to take this serious, and particularly from my point of view, looking at what is in our food and how we can be able to advise, consumers to be able to cut down on their risks of being exposed to food contaminants that can contribute to neurodevelopmental disorders. Let me focus on one of these food contaminants, mycotoxins, keeping aside heavy metals, bisphenol A, pesticide residues that we also look into in our group. So mycotoxins, first of all, are secondary metabolites produced by toxigenic fungi species that easily contaminate our food. They are invisible, they are colorless, odorless, tasteless, they are stable and resistant. For example, we would require temperatures higher than the average cooking temperature or the normal cooking temperatures in Africa to be able to destroy mycotoxins. For example, aflatoxins, we would require at least 350 degrees Celsius, but cooking temperatures in Africa traditionally do not exceed 100 degrees Celsius. So how then do we face this invisible and colorless contaminants? So to give you a bit of background, mycotoxins are produced by fungi, and fungi tend to migrate with their colony from one place to the other when they feel threatened. Unfortunately for us, this fungi migrates and settle conveniently on our food. And when they sense dangers around our food, they turn to secrete secondary metabolites. These are the mycotoxins, which are protective to them. But unfortunately for us, they are toxic for us. And they are secreting them on our food. From time to time, when they feel they are done with that area, that's the food they have contaminated, they moved with their colony to elsewhere. But these secondary metabolites, that is their protective substances, remain, and they are invisible, and we cannot see it. So sometimes you find a food that is actually contaminated with mycotoxins, but you cannot see the fungi, which are visible because they have migrated. But presence of fungi does not mean that mycotoxins are there. They only secrete these metabolites, these secondary metabolites, when they feel threatened to protect themselves. So a good number of these mycotoxins are known in the literature, more than 500 already, but about 8 of them are known as mycotoxins of public health importance, some of which include the aflatoxins, especially the B1 category, ochratoxins, the A category, deoxynivalenol, zearalenone, fumonizins. And interesting to say is that they contaminate the spectrum of food, and each one of them has a cream of food that they contaminate. For aflatoxins, for example, grains like corn or maize and groundnuts or peanuts are very frequently contaminated. And these are just some of the structures of a few of these mycotoxins, and in red are the functional groups that are exploited during these interactions with body components to be able to elicit or trigger specific unfavorable reactions to our health. And how do we become exposed to this? Yes, the fungi settles on our food, filtrating, secretes secondary metabolites, the mycotoxins. And when we eat this food, sometimes as humans, we eat it directly from our food. Even if we cook it, the temperatures may not be enough to destroy them. But sometimes, the animals feed from this food, or sometimes the spoiled grains are used to produce animal feed, and then the animals take it up and metabolize these mycotoxins. For example, aflatoxin B1 can be metabolized into aflatoxin M1, which are both carcinogenic and also have neurodevelopmental effects, or otherwise neurotoxic. And so far, a number of disorders have been investigated from acute exposure through chronic, including malnutrition, immunosuppression, stunting growth, liver cirrhosis, cancer, and so on, which we have also attempted looking at in Cameroon. But neurodevelopmental disorders are not that adequately exploited, and that is why our team is taking an interest in a slight shift from 2024 going forward. So dietary intake is the primary route of mycotoxin exposure in humans. Of course, cereals and nuts being the most frequently contaminated sources, and unfortunately for us, they are staple foods in Africa and Cameroon. 75 to 100% of agricultural commodities in Cameroon, by my estimation, based on our research output so far, are contaminated by mycotoxins. And 45% of cereals and nuts in Cameroon have been detected to have levels of these mycotoxins, especially aflatoxin and fumanizid, that exceeds the European regulatory limits for these mycotoxins. And this is a big problem to us. Unfortunately, at the minute, we do not have any regulations, and we are strongly working towards making proposals. And the best way is to be able to justify the impact on the community, on the consumer population, that's the nation, so that policymakers can be able to find a way to understand easily. So mycotoxins are so difficult to eliminate because of their difficulties, because they're produced by fungi that are ubiquitous. And of course, exposure is almost unavoidable, because when they find themselves in our food, we cannot, even by our cooking temperatures, destroy them. And of course, when we become exposed to these mycotoxins, you can picture here that almost all the different body organs, including the essential ones, are exposed. For example, the lungs, fumonization, may have a grievous impact, the heart, the liver, and so on. And imagine that once the liver is touched, the immune system becomes threatened, because metabolizing this, the liver being the body organ that is responsible for metabolizing different contaminants that come in, or different chemical substances become impaired, then the body is almost exposed to embrace almost everything, and that is not good for our health. So the effect of this mycotoxins, the degree of toxicity, depends on the species that has been exposed, age, sex, nutritional status, and all the associated disorders, but also include the different types of mycotoxins that the body is exposed to, and the extent to which this exposure occurs, and the initial body state or health state of the individual exposed. But permit me focus on neurotoxicity as I explained here on. Now, based on some of those literature review and the publications we have put out there, we have been able to produce this little diagram to show exactly how mycotoxins, may potentially affect some parts of our brain. For example, oxidative stress can be induced by aflatoxins B1 or protoxin A and fumonizin B1, disruption of blood-brain barrier, alteration of neurotransmitter systems, and mitochondrial dysfunction, as well as activation of immune system and neuroinflammation, which I presume have been highlighted in previous presentations. So, but what makes children in particular vulnerable to exposure to these mycotoxins generally, irrespective of the angle of the globe? Children have immature detoxification system. They have high food intake per body weight. They have frequent consumption of contaminated food. For example, in Africa, I would say most children like crawling, and they would crawl not just on clean floors, but they prefer crawling on bare ground that we are not sure of what is in there. And so they pick up things, and they have that tendency of picking up things into their mouth, or grabbing food into their mouth without washing their hands, especially when they are not being looked after by adults. And of course, they may be exposed even through breastfeeding, when the pregnant or the breastfeeding mother feeds from mycotoxin contaminated food. But the context of Cameroon, based on our experience and the experience of related researchers in Cameroon, shows that children are particularly vulnerable because they are exposed to consumption of a combination of different foods that are already prone and have been detected to have high levels of mycotoxins contamination. And of course, children in Cameroon, like in other parts of Africa, tend to feed from the same pot as adults. That means that the food that the children would eat actually may expose them to contamination, whereas this level of contamination may be insignificant or inadequate to trigger negative effects on adults. So if we have a set of food for children being prepared, probably there would be careful thought about the ingredients and the components going therein. And of course, poor post-harvest handling and storage practices has influenced contamination of toxins, mycotoxins, and that allows food for complementary food preparation for children to be at risk. And of course, as mentioned earlier, no legislation on mycotoxins in Cameroon so far. And of course, implementation of our Virgin Food Safety Framework Law that came up in 2018, December 11th, is still very timid. And of course, low dietary diversity amongst these children, particularly low and rural, low income populations in the rural areas. And of course, inadequate public awareness and so on and so forth that we have engaged into. So in the course of our publication, which our manuscript, which would be coming up soon, we came up with this figure to be able to look at the spectrum of activities that may occur from prenatal through early stage development once an individual or a pregnant woman become exposed to mycotoxins through first, second, third trimesters of pregnancy until early childhood of the unborn baby. So if you look through this, at almost all stages, the child is exposed because the more that the child actually develops in the stomach through placental contact, food, blood, through the placenta from the pregnant woman to the baby, the mother to the baby. And of course, when the baby is delivered, the baby also maintains another contact, which is through breastfeeding milk. And it's been known and well, it's been confirmed that mycotoxins are able to go across the placenta and can also be carried over into breast milk and to the child. So if the pregnant woman is exposed to mycotoxins, the child is potentially vulnerable. And of ages below 5, the child may start suffering from low IQ, attention deficit, autism, and so on and so forth, which we are trying to go down concretely to investigate and bring potential data set. In terms of the work we've been doing in our team from Cameroon, this is just a few of some of the data set, and I have chosen to project only one of those mycotoxins, the aflatoxins, either total or aflatoxin B1 in particular. Total, it would include B1, B2, G1, G2, and in some cases M1. So if you look at it percentage positive, you would realize that the lowest of the percentage we got was 24%, but the majority of the percentages are above 75. And of course, the contamination levels are generally higher than the regulatory limits altogether. And in terms of biomonitoring studies, that means collecting urine samples or breast milk samples or blood samples and investigating the presence of these toxins after seeing them in the food that these individuals consume. We've been able to look at healthy populations. We've been able to look at people living with HIV/AIDS. We've been able to look at children breastfeeding mothers, and the urine has reported in different parts of Cameroon. We've found percentage contamination spanning from 3, which was the lowest in people living with HIV/AIDS, right up to to 73 on the apples. And if you look at the mycotoxins, M1 have been detected, and of course, levels again are higher than the maximum limits set by the European Commission, also the Scientific Committee on Food. And when we perform some toxicologic analysis, we realize that the margin of exposure for aflatoxin B1, for example, has been always less than 10,000. And this means that exposure to this contaminant is a concern and should not be tolerated. We have also found this aflatoxin B1, which is the most dangerous of the mycotoxins within the side of the globe, and also fumonisin, which is also a very toxic mycotoxins and very common in this side of the globe. We have found that their co-occurrences has also been a problem, but we are yet to investigate whether the effect is potentiation or synergistic once they find themselves in samples, blood samples of individuals that are exposed. So from the broad picture of what we have seen from literature review and put those papers out there, we move to the second work package, which is going into the specifics. So here we are interested in going down to the Cameroonian field, which we have actually done some dotted studies, but this time around looking at all the 5 agroecological zones in Cameroon and spanning across different disorders, unlike just focusing on HIV, people living with HIV or those who are suffering from cancer, but we want to span on a majority of these disorders, including on top of our list, neurodevelopmental disorders. So we intend to collect food materials that are being used for complementary food preparation for children. Some have been sampled already, and preliminary results will be shared. And of course, collect the blood samples and urine samples from these children, from the pregnant women, breastfeeding women, and also get blood from umbilical cord just after delivery, and also blood sample from the newborn. So we are actually in the process of looking at how that will happen. So to share what we have done so far, we have already begun the process. We've picked up some food samples and blood samples and complementary food directly from the plates of this on the 5 children when they were being fed. And also we've collected some feed samples because we understand that if the animals are fed with contaminated feed, the individuals, we as humans, we later on eat flesh or animal products like egg, which is common for children as a food component for children. Then we may be exposed to these mycotoxins because they are known to be carried over. So in terms of preliminary results on the field so far, we have been able to look at pregnant women in 2 ecological zones of Cameroon, and so far in their blood we have reported pregnant women blood that we screened all were positive for aflatoxin B1 with a mean level of 20.3. That's a tremendously high, because I would say here that there is no threshold for aflatoxin B1 in the blood. That means that the slightest amount is problematic, but here we are reporting 20.3. And the lowest range, the minimum value was 5.3 and up to 92.7 as maximum limit. This is a very, very high exposure amount, not to be tolerated, and we continue the investigation. City of Cameroon, we collected 36 urine samples from 36 different pregnant women at random in some antenatal clinics where they go for antenatal. All of them were positive from aflatoxins, which are neurotoxic. And the lowest level of exposure there was 19.8. And the maximum, the mean was 19.8, the minimum was 7.2, and the maximum was 24.3. This again is not good at all. So for food, the analyses are ongoing, and 3 of my master's students, Idris, Nikel, and Shimen, have been working on that. And in terms of the under 5 children, we started off with the malnourished subset, and we would be expanding. Of 77 children that one of my MSc students, Ernest, has been working on at the reference hospitals for children with malnutrition in Cameroon, the Chantal Biya Foundation, we reported 100% presence of aflatoxin in the urine of all 77 children at a mean of 7.6, and of course a minimum level of 1.5, maximum 15.8. So we basically want to expand and look at all 5 agroecological zones and collect food samples from the plates of the under 5 children, and also food samples that the pregnant and breastfeeding mothers are feeding from, and also food samples from the plates of already detected and well-known children living with autism. And then we want to also collect their blood and urine samples, and then do a large-scale screening and come up with results that are very, very nation representative and can advise policy, especially for the neurotoxic mycotoxins. But we have that limitation because so far we are just trying to manage this research from our own mega sources, and we hope we would be able to pick up a project that will allow us expand. So for work package number 3, which involves looking at the dynamics between mycotoxins and neurodevelopmental issues in the young children, we have started off looking at the animal sector and also to look at primary school children. So what we intend to do further is to be able to expose these animals to their normal feed, which we will test to confirm that it is contaminated with neurotoxic mycotoxins. We tested it before twice, and we found levels of neurotoxic mycotoxins or mycotoxins, multiple mycotoxins generally. So whatever feed that is going to be given to them, we're going to test and know the level of this contaminant there. We're not going to contaminate them with mycotoxins, but we're going to take the natural state of contaminant, know the threshold levels, the levels at the beginning, and then expose them to their normal feed, which is what they are actually going on to. They're actually being, depending on as the feed they are being fed. And then we actually follow them over a period of time, and we will be able to expose them to, feeding twice every day, and perform body weight taken every 2 days, and perform some clinical examinations. And of course, we would, during that period of feeding, follow up on their behavioral changes and record them. And at some point, we'll sacrifice them and run biochemical parameters to investigate the oxidative stress, brain neurotrophic factors, and so on. And then we perform some real-time PCR analysis to look at what becomes of their messenger RNA, especially as synaptogenesis, and look at the interaction, some epigenetic reactions, how this mycotoxins might have interplayed with some body parameters or molecules. And then we would do, of course, histopathological examination after these animals are inhumanly sacrificed. And of course, we would expand to preschool children and primary school age children under 5 to be able to see just behavioral characteristics and get the food that they eat and try to detect presence of neurotoxic mycotoxins therein if they exist. So again, I would like to stop at this point to say that this piece of work we are trying to put forth is actually an initiative of myself and our team. And I want to say a very big thank you, especially to the organizers of this Team the Innovine meetings, and all those who have made great contribution, that I know that our presence in this meeting is not just to share what we have done, but also to absorb, to learn more, especially in the neurodevelopmental angle, some at molecular levels. And the best way we thought we can learn this is to open our hands, to embrace collaborations, to embrace pieces of advice, to embrace any form of support, to collaborate with us to see that we go through with this, not just for Cameroon, but for the Central Africa and Africa as a whole. And I'm sure that this is the beginning of a very fruitful and long life collaboration. Thank you once more, Innovative Meetings. And thank you, Laura, for all the constant communication. And thank all of you for being there to listen and to be able to provide contributions. Thank you so much. I would stop at this point, Laura.
Yes, thank you so much, Wilfred.
Thank you.
Thank you for your wonderful presentation. It was very engaging and informative. If you have any questions to Wilfred, please unmute and ask him directly, or you can post them in chat so that he can answer your questions.
Dr. Abi, I just want to thank you for this incredible work and research you're doing with your team. That's great work, to be done in Central Africa. So congratulations. I'm just wondering, because I'm more interested in the cultural aspect, is how families relate to or contribute their kids, weakness to evil spirit possessions or magic. How do you deal with this? Have you mentioned in your presentation that some families or parents they contribute to their kids' sickness, or when they become unwell, physically unwell, to demons, to demonic, energy and evil spirits. That's a common thing in the Cameroon. And how do you deal with it? If you come across, patients or families with these, attitudes. Thank you.
All right. Thank you so much for this very insightful question. So typically, actually one of those nations that is multicultural, and in different angles, they have different beliefs. So generally, in some particular communities, they would actually know that it's abnormal for this to occur, and that it would have been done by someone through witchcraft and things like that, mystically. So what we do when we come across such situations, of course, we are still at the preliminary stage, but then we have been able to speak to some parents of some children. And the first thing we put forth is that we know this is not what any parent would want for his or her child, but it has occurred. And from our own point of view, we are researchers, and we have been reading a whole lot of work that has been done across different parts of the globe, and we know.
You've been cut off, Dr. Abia. Your internet is, I think,
It
playing
would
up.
be important for us to know, first of all, that these contaminants we already know that are capable of inducing this kind of disorder that your child may be suffering from may be present in your food. So we would like to invite you to collaborate so that we can first of all investigate your food, and then get to speak with the child just to understand whether the child has that impairment and to what extent. So once we do that, we collect the food, go to the lab, and it keeps the parents, first of all, in a call, because when it occurs, know that medically it can be through research, as I mentioned. If parents, if the population knows as much about their food and how their food interact with their health and the environment, they can be able to take their health into their hands and reduce exposure to contaminants that may lead to those kind of disorders. So once the parents cooperate, that is a very big step. And the next thing, we try to find out if the parents have been able to get diagnosis from the hospital and confirmation and advice from the medical personnel. Where they have not been able to, we encourage them to do that. Of course, we have a center for handicaps here in Yaoundé, where we have children also that are suffering from autism, and that our part of our research, we've started negotiating how to actually follow up some of these children. And our hope in the greater scale is that when we are able to convince some of these parents scientifically, in addition to the medical advice that medical personnel have given to them, we would, at the end of this project, during capacity building, encourage those parents to to give life in other people who may be suffering, whose children may be in the similar circumstances, by sharing their experiences, their beliefs at the beginning, how they went about it, and how they got to approve of medical outcomes and also research findings, and how this has changed their whole point of view. Thank you very much.
Thank you. Great work. Keep up the good work, Dr. Abia.
Thank you so much. I
Do
appreciate
you
it.
mind if I make a compliment? You're dressed very cool. I like your outfit. It's so cool.
Thank you so much. Thank you so much. Thank you so much. I really appreciate everything.
Dr. Abia, great research, really
Yes.
interesting. I have a question. How, when you go and talk to those parents and you educate them about the food, what is the affordability of the food that is, let's say, cleaner for those families and actually for most of the population in Cameroon?
Thank you very much for diving into this aspect of the discussion. I would begin by saying that a couple of years in the past, Cameroon has been known as the breadbasket for Africa. And over time, something happened, and Cameroon is almost losing that. Since the 90s, we started losing that. And along the line, we first of all brought in a lot of pesticides, and there was a lot of residues that have been detected, and we're still following that up. And the government takes a lot of time to try to polish up some of these things and to be able to make sure that we get back by introducing agricultural initiatives for farmers and encouraging people to go towards farming, which is very good. In terms of affordability, at the present moment, it is still very challenging. And it is part of the reasons why we also have contaminations of our food by these neurotoxic contaminants. First of all, climate change is not giving us any space, and we continue to wrestle with it. Secondly, economic hardship of the population does not allow us to conveniently eat the kind of food that people want to eat, because in some areas, in some instances, people actually will eat what they see. So we have that slogan of there seem to be more mouths than there is the food that is available and accessible in a sustainable manner to be consumed. So we try as much as possible. This now, it comes from an NGO that I head as well, Integrated Health for All Foundation. We try to encourage such communities and such people or communities in this kind of circumstances to go towards petite agriculture, petite farming, home gardenings to produce some of the things, to cut down on eating the things that they did not produce or do not or hardly know where it comes from, especially in their local communities, because it was never the case. And this triggers another component, which is the food fraud component, that we are finding it exposing and releasing a lot of contaminants in the food intentionally by food business people in what we call food fraud. And most of these contaminants also contribute to these neurotoxic effects. So that's another school of thought that we are also following in bits. But then I would conclude that the government's initiative, which if the citizens apply, follow it, to an extent, we would be able to overcome this issue of food security, not having enough affordable food available to us to consume. And that, in a way, in a bit, can help reduce the challenge, can reduce our exposure to these contaminants. Thank you very much.
Thank you so much, Dr. Abia. And I hope, for Cameroon, for the whole world, that our kids, our pregnant moms, ourselves, that we can all have access to clean food, clean water, and air. So thank you for the great research you have done.
Thank you
Or
so
are
much.
doing.
Thank you so much. And I appreciate all the comments and the opportunity so far.
Thank you. Thank you so much, Wilfred. Any more questions for Wilfred? Okay, thank you, Wilfred. Thanks once again for your expertized information sharing with our audience.
Thank you so very much. I will now stop sharing my slides. Thank you so much, and I appreciate it, and I'm open to collaborations. Thank you so much in advance.
Yes, next I would like to invite Jehanna Andrews from Western Sydney University of Australia to present her talk on mental health issues affecting immigrant women.
Sorry, give me a second. I need to make sure I'm sharing the right screen. 1 second, please. Thank you.
Yes, sure.
Mhm. Okay, I guess we'll get started. I don't want to hold you back.
Yes, Jehanna, your slides are visible now.
What is it? Okay. Okay, welcome everyone. We'll just keep going. I'll be talking to you about the mental health issues affecting immigrant women. My presentation is not as sophisticated as Dr. Ian. So, yeah, I'll just start then now. Gender has been recognized as a significant factor affecting or impacting on the mental health and well-being of women. The combination of women's biological, cultural, economic, and social roles, responsibilities, and expectations puts them at a risk of developing mental health problems than men. Women's mental health has been identified as one of the top 5 research priorities worldwide, and the risk of women developing depression and mood disorders is twice that of men. This risk is even higher among immigrant women due to cultural factors and issues related to migration and settlement. While many women who migrate settle quickly, others do not. For some, migration can be a traumatic experience. Migration is one of the contributing factors for depression, anxiety, and suicide. Australian studies, which is also quite similar to other international research, found that suicide rates for many immigrants were higher than those of the population in their country of origin. And they attributed this to acculturative stress and issues of migration. So why is women's mental health affected by immigration? This has to do with difficulties in adapting into a different culture due to the grief over the loss of their previous homeland, over the separation from family and friends, isolation, language barrier, lack of social support, and also it could be related to pre-migration stress. For cultural reasons, many women take little part in decision-making. They migrate simply because their parents decide it's to the advantage of the family. On arrival to the new country, the young immigrants would assist their parents in managing their day-to-day issues, appointments, interviews, and act as interpreter, which add an extra stressor or responsibility towards their parents due to language barrier. Immigrant populations commit 25% of suicides, and 60% of these are committed by younger groups, with more suicidal behaviors in girls. For the immigrant young woman who's striving to develop a satisfactory sense of identity, like any other adolescent, may also be faced with extra stresses related to immigration and cultural conflict. She may not have had a choice about the decision to migrate, which results in a process of loss and grief, which may resolve over some time or may continue to create emotional problems for the young woman. The suicide rate is even higher among refugees or young refugees in particular. There are issues related to domestic violence, sexual abuse, which increases the young person's vulnerability. There is also bullying at school, which can also impact negatively on the mental health and well-being of the young migrant or refugee, as well as causing deterioration in their academic performance. It can lead to also eating disorders. Also, issues related to discrimination and language barrier can also be an obstacle to, even if they wanted to access mental health services, it acts as a barrier. Many young migrants and refugees have experienced traumatic events in their home countries, such as war, violence, and persecution. These experiences can lead to mental health issues such as post-traumatic stress disorder, anxiety, and depression. Globally, about 12 million girls each year get married before the age of 18. In Australia, 83% of one-parent families are single mothers, and women do over 9 hours a week more unpaid work and care than men. In Australia, 23% of women compared to 8% of men age 18 years and over have experienced sexual violence in their lifetime. And that's not just in Australia, that's actually globally and, internationally, including childhood, sexual abuse and or sexual assault since the age of 15. About 53% of the perpetrators of sexual assault against women were intimate partners, and 69% of the incidences occurred in a home. It's estimated that children below 18 years constitute 50% of the refugee population worldwide. Due to conflicts like the Syrian crisis, children are continuously exposed to traumatic events. Trauma exposure can cause mental health problems that may in turn increase the risk of morbidity and mortality. For refugees, which is a different category compared to people who migrate voluntarily for a better life for themselves and for their families, acculturation occurs within a context of forced change. They experience trauma related to the sudden separation from or loss of family, and the effects of torture and trauma experienced in the country of origin. They are faced with the challenges of and post-migration stressors, including discrimination and economic hardship, integration into the country of resettlements. Torture may take many forms, such as deep sleep deprivation, isolation, beating, burning, starvation, mutilation, being raped or brutalized. 1 to 2/3 of refugee women seeking treatment as a result of torture reported sexual violence. This sort of torture fosters dependency, debility, and dread. That's the 3Ds, which together will destabilize and dehumanize the victims and entrap them in a situation which destroys their psychological and physical resistance, self-image and identity, and loss of trust, all of which will have a negative impact on their mental health and well-being. Women are more frequently the victims of violence. Fear and trauma dominates the lives of many young women and significantly affect their mental health and well-being. Sexual abuse is as much a problem for young women as it is for all women in the home and social situations, including workplace. Research has shown that called women do not report sexual harassment or abuse because they feel ashamed and consider it reflects badly on them and their reputation in the community. Violence can take the form of physical, social, financial, emotional, psychological, and sexual. Reports highlight that the prevalence of domestic violence or family violence is high for all women in Australia. And again, I would say it's not just in Australia, I would also say worldwide. However, research is showing migrant and refugee women, including younger girls, are more likely to be impacted by the consequence of domestic family violence and the barriers to seeking help. The ABS shows that an estimated 8 million Australians have experienced violence in the form of physical and or sexual since the age of 15, including 31% of women experience physical violence and 22% of women compared to 6.1% of men experience sexual violence. So both genders are exposed to sexual violence, but it's more prevalent among women, and that's not a surprise. It's been always like that anyway. Again, that's international statistics. 1 in 3 women are exposed to domestic violence or some sort of violence, whether it's a combination of physical, emotional, social, financial, or sexual during their lifetime. And this occurs regardless of age, gender, race, sexuality, wealth, or geography where they're living. However, there is an extensive body of evidence that identifies how gender and power relations are unequally distributed between men and women, and that this is the primary contributor to domestic family violence. This is especially true if the woman, is earning more money and bringing more income, more than, her partner. This can create, family conflicts, especially in male-dominant societies. Some women get married at a very young age in an arranged marriage, which again, they may have not necessarily have an opinion or a choice about whom they want to spend the rest of their lives with them. In some religious groups, some women are forced to wear the hijab and forced participation in religious activities. Reproductive abuse again was reported that their intimate partner was the perpetrator of physical violence during pregnancy, rape aimed at causing a forced pregnancy or a miscarriage, and in some cases forced them to have an abortion. There are further contributing factors impacting the experiences of domestic family violence among women from refugee and migrant backgrounds, including language, religion, race, ethnicity, and class experiences that need to be understood and acknowledged in relevant research and policies. Perpetrators have their own tactics to hide, destroy, or control access to their partners' mobile phones that women relied on to access support. They may prevent them from seeing their friends or their other family members. Again, in some cultural groups, the family plays the primary source of, providing emotional and sometimes financial support to these women. So the perpetrator is trying to cut women off from their source of support. Again, these issues was raised when the woman is dependent on a partner's visa. Sometimes he would make threats. I'm going to send you back home to your country, or I'm going to report you to authorities, and making threats. In some religious groups, the values of patience, endurance, and forgiveness are often prioritized over safety. The research highlighted the patriarchal expectations of family life and unfortunately, reinforcing domestic and family violence as a private matter, and that if women sought assistance for domestic violence, they would not be believed. So women just suffer silently because believing that no one would believe them. And that's part of a religious obligation that the women need to endure and persevere as part of a religious or spiritual matter. Culture plays a role in the way men and women and society in general interpret what constitutes violence, what is acceptable, what is not acceptable behavior, which behaviors can be tolerated or not, and what constitutes severe or mild forms of abuse. Diverse views from newly arrived communities in relation to what constitutes domestic and family violence and gender roles. Especially I'm talking about new immigrants. They don't know much about the rights and the protective laws that protect women, and also the support services available to them. There is a key knowledge gap in the understanding of domestic violence, which limited, because most often they limit domestic violence, or their understanding of domestic violence or abuse is when their partner hit them physically or abused them physically. They don't understand that also emotional, social, financial, and spiritual abuse are considered the severe cases of and unacceptable behavior. And they considered, other type of abuse as perceived as normal gendered roles. Victims of domestic violence, when compared to others from nonviolent homes, have higher levels of stress, anxiety, depression, and other psychiatric conditions. And, there are 5 times more likely to attempt suicide, to have higher rates of alcoholism, to be 9 times as likely to abuse drugs, experience sleep disturbance, reduce coping and problem-solving skills, and to be socially isolated. These problems represent major impairment of women's mental health and well-being. And that's a vicious cycle. More likely that these women, or if they're parents, if she's a parent, she'll also abuse younger kids. Young women who experience domestic violence may also may face compounded mental health challenges such as complex post-traumatic stress disorder, adjustment disorder, intergenerational trauma. The trauma experienced by parents can affect their children. I just mentioned this, leading to a cycle of mental health issues. They may also carry the emotional burden of their parents' experiences, which can impact their mental health and well-being. And also there is a fear of authority figures. Survivors of domestic violence might develop a fear of authority, including healthcare providers and law enforcement, making it difficult for them to seek help. That's another big issue here, intergenerational and intercultural conflict. Research links these conflicts to increased self-harm and suicidal behavior. Acculturative stress contributes to emotional challenges such as depression, anxiety, low self-esteem, identity confusion, isolation, and anger, long-term distress, and feelings of powerlessness. Some young people respond with rebellious behaviors, including leaving home or taking drugs as a way to reject parental authority. The existence of double standards between the sexes within traditional cultures, which permit males greater social freedom, independence, and educational opportunities, but places greater constraints on females, is a source of conflict and contributing factor for eating disorder. Recent Australian research also indicated that immigrant young women from both Mediterranean and Asian backgrounds are now developing bulimia and anorexia nervosa, a disorder uncommon in their country of origin. And the researchers suggested that this is related to long-term unresolved acculturative stress. There is issues of discrimination and racism, which contributes to social isolation and exclusions. We've mentioned about the intergenerational conflict when the young ones trying to navigate between parental values and the host culture norms. Language barrier, limited proficiency in the host country's language can hinder young immigrants and refugees' ability to access mental health services, communicate their needs, and integrate into the community, as well as schooling issues, including bullying at school, because of their accent or because they cannot speak, they're not proficient in English. It's also like because of language issue and culture difference, the young one may have difficulty in maintaining peer relationships, especially among children less, under age 15. Language barrier and schooling challenges are also linked to emotional distress, self-harm, and suicidal behaviors in immigrant youth. Of course, there is a big literature on the impact of bullying whether at school age, or bullying at work, or bullying wherever you are, in the development of mental health, issues, whether you're an adult or young person. Of course, the younger ones are more vulnerable, simply because of their age, being vulnerable, still their brain developing, and their emotional wholeness is still in progress in the development stage. So they can be quite or more vulnerable, affecting, of course, their quality of life. Financial instability, issues related to access to education, and poor living conditions can contribute to the mental health challenges faced by young immigrant and refugees women. If you belong to the LGBTQA plus community, that's even an extra stressor in itself, especially within traditional, classical cultural groups. There is double discrimination based on ethnicity and sexual orientation. Many refugees have endured violence, persecution, or abuse in their home countries due to their sexual orientation or gender identity. And this can lead to higher rates of mental health issues, including post-traumatic stress disorder, depression, anxiety, and suicide. There is a big literature, especially maybe in the last 10 years or so, on the discrimination LGBTQA plus communities face, even from healthcare professionals. I'm a mental health background, including mental health, professionals, which can lead to their suicide. The stigma, again, and fear of rejection from both ethnic and LGBTQA plus communities still exist. Accessing mental health services can be challenging due to cultural, financial, and systemic barriers. Sorry, pardon me. Many refugees and migrants also encounter healthcare providers who lack training in LGBTQIA plus and affirmative care. There are stigma related to mental illness across all cultures, but it's highly stigmatized. Mental illness is highly stigmatized among specific cultural groups. And there is misperception about the causes of mental illness. And based on this, people, how they view mental illness or the causes behind mental illness will actually direct them on how they can manage it. For example, some cultural groups attribute mental illness to the evil eye or witchcraft or spiritual evil spirits possession. So based on these perceptions, they will resort to maybe religious leaders or healers, traditional healers, to carry out certain, religious prayers or rituals, holy water, the use of holy water, certain prayers, and so on, or bringing in like a religious leader to carry out, an exorcism, believing that this is just demonic energy. Others may resort to a magician, go back to a magician, someone or witchcraft to revert the spell. Sometimes they would say, you hear them saying, this woman put a spell on my son or daughter to become mentally ill. So they go resort, they don't seek professional mental health for their mental health, issues. Of course, this, causes delay access to proper interventions, and we all know the importance of early identification and early interventions in speeding up the recovery of the person with the lived experience, the person with mental health issues. There is also like religious factors. Again, big literature, especially in the last like maybe more than 10 years, I've read lots of literature looking at the impact of religious leaders as a barrier to accessing mental health, like services, because they contributed to it's a spiritual thing. So they have to go to pray more, to have more stronger faith, which of course we know the importance of spirituality in recovery, especially not just from mental health issues, but also from physical health conditions. Yep. There's been some literature talking about the lack of religious leaders, collaboration and cooperation, and lack of understanding about the importance of referring people with mental health issues to proper mental health services. So it's very important that we work collaboratively with the religious and community leaders through maybe raising awareness, providing health education sessions, with them. Again, in some cultural groups, they have so much trust in the religious leaders. So whatever they tell them to do, they just do it as a matter of trust. So it's a good venue for us to explore and to do some work with community and religious leaders due to their influence in help-seeking behavior and in influencing communities' attitudes and perception and understanding. Again, there is some issues or barriers to accessing mental health services beside the stigma, beside the culture difference, issues of trust and confidentiality. Again, especially for clients or people who come from refugees, who are refugees because of the torture and trauma and fear of authorities, it stopped them from, having trust and so on. Lack of same gender interpreters prepared to work with called women of all ages, especially those women experiencing sexual assault and or with the queer communities or clients. Mental health services, as much as they try to provide culturally sensitive care, and there are lots of even becoming compulsory cultural training for all staff, there is a lack. Lots of research done, especially more of a qualitative research, interviewing, people with lived experience, their carers who come from cult background, and they report that they're dissatisfied with the quality of care they're receiving from mental health professionals across the board, from nursing, the doctors, psychiatrists. So we need to also, it's not enough to provide education or even making education compulsory. It means nothing actually to me. I'm an educator myself. Making education compulsory, maybe you will remember 10% of what you've heard from the presenter or the teacher or the educator, more of experiential learning, opportunities, what if scenarios. It's becoming more realistic, and I think it's a good way to teach, like adults through maybe using simulation in the form of role play. And that's one of the presentations I'm gonna talk about. I think it's my next presentation, using simulation to promote cultural empathy among students and healthcare professionals. To conclude, sorry, am I over the time? That's my last slide. Women from all cultures have specific mental health needs across the lifespan. So the way forward in women's health requires addressing the much broader range of factors influencing women's mental health, such as the social, environmental, economic, cultural, and biological factors. Any attempt to improve young women's mental health must take into account the importance of equity, social justice, and empowerment of women in improving their mental health. There's also a need to provide the provision of support across employment, education, welfare, social, and health sectors is a crucial protective factor in promoting resilience. There is a need for preventive measures and early interventions, early detection, prevention, health promotion, health education, community awareness, to detect younger women who are at risk and providing early interventions with social support and counseling. Thank you. Is everyone still with me?
Absolutely. Oh, this
Yes,
is
great.
it's it's a great presentation with a very difficult topic. And thank you for creating more awareness.
Thank you.
It kind of breaks my heart that that still, is an issue in nowadays, so prevalent. Mm.
And you know what? The worst thing that it's a bit, I don't know what to say. I've done similar presentation more than 10 years ago on the same topic on domestic violence. And very similar when I was looking at the new relevant literature, nothing changed. I said it made me reflect actually. So where are all these services that, catering for these, women who needs help in all different aspects of their lives, and still the prevalence is still the same. It hasn't changed over the last 10 years. It just made me stop and reflect on it.
Oh, absolutely. Absolutely. I think it's, as you're doing, is like creating much more awareness, more education, having stakeholders, the government, more research on women, understanding that our needs and our physiology, pathophysiology is quite different than men. And then how can we support women in their process? How could we prevent these huge disparities and address the gaps?
Absolutely, especially like women, historically, even in our Western cultures, historically, they grew up to be submissive, having not much say. But like due to globalization and the internet and Facebook and social media, I think women are becoming more empowered, but not all women are the same, for all the different reasons, and their personal life experiences can have an influence. I am aware personally that having a high education or a high status job does not prevent violence from happening or stop women from being exposed like to domestic violence. In one time in my community, I was doing a mental health, education session within a community, and I was surprised. I was shocked, really. Few women opened up when I start talking about mental health, and I touched on domestic violence and the different types of abuse. Again, like exactly what the literature was saying. Lots of women from cult background, they only think of domestic violence or abuse. It's only when my husband or partner hit me or bash me or something, more of physical, abuse. They have no understanding at all about the emotional abuse, which is worse than the physical. Its impact is actually worse than the physical, abuse, isolating them socially. That's huge, a big, have psychological impact on them. When you're locked up in your room, you're expected to work inside the house and outside the house, looking after the husband, looking after the kids, even sometimes looking after your elderly parents. And after this, you get abused financially. The woman go out to work, and the husband will pick up the income. I had one woman from my community. She was telling me, I don't know how much I earn. Although she was a highly educated woman, I was surprised, really. I said, it doesn't mean you're in high professions and highly educated people that you wonder or it never crossed my mind, like, like, she doesn't even know how much she's earning, because she told me the income just goes straight to my husband's account. Shocking, like, to me. But there are lots, that's only within one small community, that, they opened up to me, due to, again, to the trust. And, like, I used to regular, like, mental health education. Like, I was invited to actually do a few talks and sessions with them. And, it's just an eye-opening for me. It's different when you read about it in books and research, and published papers, or government policies, or it's different when you're actually faced with it. It's a reality. And it's, and, like, people don't report it. Women just suffer
Yeah.
silently, of fear of maybe breaking the family, and their partner usually threaten them. If you go to the police, I'm gonna kill you, or I'm gonna take the kids away, or I'm gonna kill your kids, our kids. A bit scary. It's not okay. I think the solution, maybe we work with our own communities, whoever come from different, within a specific called background, work with the community, raise awareness, about the different types of, abuse, and the support services. Again, especially if you're a new migrant, you don't know what's available there.
Yeah, I was gonna ask you if you have, run into research, or, as you're talking to different women and communities, if there has been any, let's say, community efforts, governmental, efforts, whatever the effort is coming from, creating strategies to mitigate these gaps. What had been those, and if that, let's say, has decreased the amount of domestic violence?
Increased,
So,
you mean
or
decreased, or
no, decrease. So, for example,
oh,
as you were saying, creating financial independence in a sense, or stability. Therefore, if the woman gets threatened to, or is hit, and has to stay in the marriage because sorry, okay, I'll start my video. Because of different reasons, cultural, the fear of being by themselves. But then if they have some resources, some community resources, where there is going to be, like, there's a sense of purpose, but also a sense of independence, like they will have a salary, they would have money to produce, to provide to their kids. If that, has created a difference in the, in the amount of domestic violence.
There are services, I'm aware of it, like, available, lots of services available, for survivors of domestic violence, financially, provide accommodation for them, shelter, and the employability, like support. That's when you get the social worker input, especially when they come across, to our mental health service, seeking treatment for their mental health issues. But as I said, not all women, especially if you're a newcomer, to
Yeah,
the new,
and
don't
and my
know.
question, it's more like in the prevention side. Like, let's see, is there any way that we can modify, let's say, women's education and girls' education
The
that can
problem,
empower? Go ahead.
yeah, no, please, you finish first, please. Sorry.
No, I'm just, as I'm just as shocked and saddened, and about the, the research hasn't changed in years, and in many areas of the world we're going backwards, even more. So I'm like, what had been some strategies that had been successful in empowering education, empowering self-esteem, empowering being outspoken, so that the women can advocate for themselves, but and be safe.
I've been thinking about it, and I also read it in the literature. Especially if you are not just an immigrant, if you're a refugee coming, on temporary visas, or it's not a priority. The mental health of people in general, whether male or female, they're just in this survival mood. They just want to settle in, worrying
Yeah.
whether they be given like a visa to stay in the host country, or they send them back to their country of torture and trauma, or. So not just their mental health and physical health is not a priority at all to them. Which I guess that's why it's important to talk at an early stage, because these people come without no knowledge, like tabula rasa, like it's like they're here, in a survival mood. They don't know their future, what's gonna happen to them or their families or loved ones, or they're gonna be deployed, so it's good, I think, if we target these people when they just arrive in the host country. I'm here in Australia, but probably some of you coming from other countries that host and welcome refugees and migrants. And especially again with the language barrier, sometimes migrants are in a better situation because maybe they come as for employment. So it's a requirement nowadays to migrate to be proficient in the English language. But again, I'm aware also that upon arrival to the country, to the host country, they offer them language classes. And I would think for refugees, it's offered for free as well. But again, the bigger picture, let's be realistic here. It's good to be talking like from the ivory tower. There are lots of policies in place, but and lots of services available. But how to know? In the first step is we need to give them knowledge. Knowledge is power. When they have knowledge. And they can speak the language, they can communicate. If we have translated, interpreted material, information, which there are some, I have to admit, there are some translated, documents around. Wherever you go to a community center or a migrant resource center, you find lots of pamphlets, in different languages. It's there, but again, it's not the refugees' priority or the migrants' priority. To think about violence or domestic violence issues, especially if they come from a traditional cultural groups and collective societies where the family is so important. So that which makes even women, if she got the knowledge and she knows about her rights, etc., and highly educated, she may still continue living in this abusive relationship just to protect the family, because that's how she is brought up within this cultural and also / religious, context.
Totally. Yes, yes, yeah. You're absolutely right.
Just have
Oof, such a tough.
tough. Sorry, guys, I should have warned you.
No. It's as you said, knowledge is empowerment.
Let's look at the good side. We're here to help these women, empowering them by knowledge. So if you're bilingual, you can even talk within your own small communities. When I teach my students here, I tell them, start. People get scared of people with a lived experience because of the lack of knowledge. Start within your family, friends, neighbors, your shopkeeper that you do your grocery from. Talk to them, raise their awareness. If you go to church, a mosque, a temple, talk to the people. You got the knowledge, and knowledge is power in itself. Enough of me, sorry. Thank you.
Yes, thank you so much, Jehan and Andreas, for your wonderful presentation. Your insights were both engaging and informative. Next, moving forward to the next presentation. Next, I would like to invite Omar Gabora from Speech and Language Pathologist Kuwait to deliver his presentation on the correlation between speech therapy and neuroscience from the perspective of a speech therapist. Yes.
Hello.
Yes.
Hello.
Yes. Hi, Omar. Please start your
Yes.
presentation.
Okay, I will share my screen. Can you see the screen?
Yes, perfect. Continue.
So good morning, and thank you all for being here. My name is Omar Gabura. I'm a Lebanese speech and language pathologist, and today I'll be talking about something that I believe is foundational to our work as speech and language pathologists, and that is the powerful and dynamic correlation between neuroscience and speech therapy. Understanding how the brain works is not just interesting, it's essential to how we diagnose, plan, and carry out effective therapy. So we often think of speech therapy as working with articulation or helping people find their voice again. But at its core, speech and language therapy is about helping the brain to communicate better, whether it's with words, sounds, gestures, or devices. In this presentation, I'll be walking you through how knowledge of neuroscience informs our clinical decisions, uh, on how we treat both adults and children, and how modern approaches are rooted in the brain and science. So as a brief, I'll be explaining how neuroscience informs speech therapy practice. We'll be highlighting the neurological basis of speech and language pathology. And finally, I will be showing practical implications about treatment and assessment when it comes to neuroscience. So let's start with the big picture. Why does neuroscience matter to us as speech and language pathologists? Because everything we work on, including speaking, understanding, reading, writing, swallowing, is all managed by the brain. So when a patient has a stroke, when a child has autism, when someone develops Parkinson's, the core issue is in the nervous system. So to treat communication effectively, we need to understand the underlying neural basis. This connection helps us choose evidence-based this connection helps us choose evidence-based interventions and anticipate challenges that are based on neurological conditions that we are dealing with. So let's take a look at the main players in these terms. So speech and language pathology involves a network of brain structures, starting with the Broca's area, which is responsible for speech production. So when this area is damaged, as Wernicke's area, when this area is damaged, we can see a patient that has a non-fluent aphasia which is occurred by a stroke in the left hemisphere. As for the Wernicke's area, this area handles language comprehension. So damage here leads to fluent speech, but it could be described as nonsense as well. The motor cortex sends messages to the muscles involved in speaking. As for the auditory cortex, it helps us process and differentiate sounds more effectively. So finally, with the cerebellum and the basal ganglia, these are like the key factors to fluency, prosody, and coordination. But most importantly, all of these areas must work together in order to have an effective communication. However, it's not just about the areas, it's also about the pathways. To speak a single word, your brain has to plan the movement, send the command down the cortical tract, and activate the appropriate cranial nerves. If the message does not get through, the muscles won't move correctly. This is exactly what happens in motor speech disorders like dysarthria or in conditions like ALS. Understanding these pathways helps us identify whether the issue is cortical, subcortical, peripheral, and that's critical when planning effective therapy plans. Now, you can consider that there are good news in terms of neuroplasticity, which is the ability to change, or the brain's ability to change. And that's foundational in our therapy as speech and language pathologists. So neuroplasticity is the brain's ability to recognize, to form new pathways, and to compensate for the loss of function. Every time we repeat a word, we practice a sound, or we model a new behavior, we are compensating for loss of function. And we are making the brain rewire itself every time we do the same process. And that's why repetition, intensity, and function relevance are the cornerstones for effective therapy. let's shift for children for a moment. In early childhood, the brain is rapidly developing. The first 3 years are especially important for language acquisition. If there's a disruption in this window, like in developmental language disorder, autism, or childhood apraxia of speech, the neural systems that support communication may not form correctly. But then again, thanks to neuroplasticity, early intervention can have a huge difference. That's why our work with toddlers, preschoolers, and young children is not just important, it's time sensitive. So if we want to connect some common disorders to their neurological roots, we can start with aphasia, which is caused by damage to the language centers, usually after a stroke. Also, we have dysarthria, which happens when the brain cannot properly control the muscles of speech, often due to conditions like cerebral palsy or multiple sclerosis. Moving on to apraxia of speech, which is a motor planning disorder. The brain, in this disorder, the brain knows what it wants to say, but can't send the right instructions to the mouth. We also have fluency and voice disorders like stuttering or vocal tremors, which have ties to neurological regulation. But what's important is to understand the origin, which helps us choose the right tools, goals, and therapy styles. Moving on to neuro-based therapy approaches, many of most effective interventions are directly informed by neuroscience. For example, melodic intonation therapy taps right into the right hemisphere by using musical processing to support language and aphasia. Moving on to prompt therapy, which gives tactile feedback to reinforce motor planning. Also, we have the constraint-induced language therapy, which pushes the brain to use the verbal output instead of using gestures or compensation. This strengthens the neural connections through forced use. Finally, we have AAC, or Augmentative Alternative Communication Systems, which are based on understanding the cognitive load and brain access to visual and motor memory. So I'll be sharing a quick case study of a 55-year-old man who has a left hemisphere stroke that left him with Broca's aphasia, as mentioned before. The conditions that he had or the symptoms were effortful and halting speech, but his comprehension is relatively intact. We used MIT, which is melodic intonation therapy that I have mentioned before, starting with simple rhythmic phrases. That's because the singing engages the right hemisphere areas where we were able to tap into the undamaged regions to support speech. Over several months, his expressive language improved significantly. This case study serves as a clear example of how neuroscience and speech therapy can actually result in a huge change in a person's life. Moving on to pediatric therapy. In pediatric therapy, neuroscience also guides on how we engage children. In children with autism, for example, we know that there might be differences in the mirror neural system, which affects imitation and social communication. So we use strategies like modeling, joint attention, and sensory-rich activities to stimulate the brain's ability in the right way. For kids with CP or cerebral palsy, we adapt therapy based on their motor planning abilities and their neural tone. What is important to understand that each strategy is grounded in an understanding of the brain's development and response to intervention. And now for technology and neuroscience, it's important to understand that technology has given us a window into the brain like never before. For example, functional MRIs helps us see which areas are affected during speech. As for EEG, it allows us to measure the timing and activation. Moving on to diffusion tensor imaging, which shows us how white matter pathways connect language areas. These tools do not only help researchers, they also guide clinical decisions, especially in complex areas or complex cases. Looking forward, we're entering a world where brain-computer interfaces, neurofeedback, and AI might become a part of our therapy or clinical-based decisions. That said, we also need to be cautious. Neuroscience is a powerful tool that we can be used, but it's not a magic wand. Every brain is different, and not every therapy works the same for every and each individual client. We should always balance science with compassion and data with functional relevance. And most importantly, we must always individualize treatment, as neuroscience gives us direction, but the client gives us the call. So here are a few takeaways or a few take-home points. That speech and language are brain-based. That's why neuroscience matters. Understanding the brain helps us be better diagnosticians and better therapists. Neuroplasticity is real, and that's the reason our clients tend to improve. And finally, staying informed about neuroscience keeps our practice aligned with the best and most effective care we can offer. I'll be ending with this quote, as I feel it truly captures our work. To treat communication, we must understand the communicator and the brain that makes communication possible. Our therapy is not just speech, it's about the systems in the brain that allows us to connect, share, and be human. Thank you for your time and attention. I'd love to hear if you have any questions or if you can share how your experiences are interlined with neuroscience. And thank you for listening.
Yeah. Yeah, thank you so much, Omar. Thank you for your wonderful presentation. It was very engaging. And I would like to invite audience for any other questions to Omar.
I would have a question for Omar.
Yes,
I'm
please
Gabriel. Please tell me, from your experience, there is, from your knowledge, there is an area in the brain in which we localize the language, or is the entire brain?
Yes, so both like as I mentioned before, there are certain areas that affect the brain's acquisition of language in terms of comprehension, in terms of expression, and as I mentioned, that the most affected areas are Broca's areas in terms like we have aphasia. That's why like it's called Broca's aphasia. There's also Wernicke's area. There is the cerebellum, the basal ganglia. So each area is responsible for a certain language aspect. And that's how it affects like the language part. So for example, if we have a stroke in Broca's area, which is the left anterior part of the brain of the frontal cortex, we can like have our communication is based on accurate comprehension, but the expression is truly affected. As for Wernicke's area, it's the complete opposite, in which we have like our comprehension is weak and it's damaged, but our expression remains like intact. But due to weak comprehension, we tend to have good speech that is like clear, but it's nonsense in terms of we speak. So I hope that I answered your question.
Okay, thanks.
Omar, I have another question for you, if that's okay. This is Jihan from Australia.
Hi, Jihan.
You're talking about children. How about if an adult with like in an adult agehood,
Yes.
possible to fix their speech if there's like I know a case of someone who got dyslexia, but that's
Yes.
different.
No, it's technically the same as I mentioned before, or the case that was mentioned in the presentation. It's a 55-year-old man. So our course of work is not only with children, it's with adults. But what we tend to do is, due to having like neuroplasticity is often at the age before 4 years or before like from the age range between 3 to 7 years. So neuroplasticity won't be helping us in these terms. However, we tend to work with cases who are adults. Most of the cases who are adults usually suffer from stroke in the left hemisphere or in the right hemisphere. So it does get better. Like we can actually treat the symptoms. We can work on effortful speech. We can work on vocal tremors. We can through neuroscience, as I mentioned before, the melodic intonation therapy about using melodic and rhythmic taps in order to strengthen the adult's speech usually helps. So to answer your question, even with cases that are not purely neurological, such as dyslexia, it's more helpful to be treated at an early age, especially dyslexia, since it could be discovered at an early age. But there are certain neurological disorders such as strokes or traumatic brain injuries or whatsoever that occur at a later age. These are purely treated at adult age. But in cases like dyslexia, autism, communication disorders, pragmatic disorders, learning difficulties, or should be treated at an earlier age because they are usually discovered at an earlier age.
But if it hasn't been discovered at an early age, it's still treatable.
Yes, but usually, as you mentioned, dyslexia, it's discovered at school age since the case is relatively teachers are usually the ones who report it as they see low grades, they see specific difficulties such as inverting the letters, low memory outcome, weak attention. All of these symptoms occur during school age. However, there are certain cases where people can go undiagnosed with autism at an adult age. So we also treat it. We tend to treat the environment more in these cases. We need to fix the environment more, how caregivers treat the adult more, rather than only working with the adult as a person.
So when you're talking about the environment, can you give me examples? What do you mean exactly with it, please?
So, for example, if I have a patient with aphasia, let's say, who is an adult, we tend to use simpler language. We need to break it into chunks rather than using complex sentences. We need to have we can use AAC systems in which the patient can tap at a certain image or use more advanced AAC such as eye gazing systems on a certain screen. For example, if the patient also has motor or has a difficulty in mobility, we can also adjust the posture and everything whatsoever.
Thank you. Good work. Great work.
I hope I answered your question.
Thank you. Yes, you did. Thank you. Thanks so much.
Thank you.
Yes, thank you so much, Omar. Next, moving forward, I would like to again welcoming Jehanna Andrews from Western Sydney University, Australia, to deliver her presentation on using cultural simulation in nursing education to promote empathy.
Thank you. Cultural competency is essential in nursing and midwifery as in any other, not just healthcare, specialties, but in any job. It's very important element, especially with the migrants, increased number of immigrants, or even refugees. Catching up to my with my earlier presentation, and the many countries are opening their doors for migration and the refugees. So it's very important and essential competency for the staff wherever they work in to develop cultural sensitivity, cultural awareness, cultural competency, and cultural confidence to actually be able to communicate with confidence with people who come from different cultural and linguistic backgrounds. And this, of course, is very important from a healthcare, perspective to provide holistic care and provide a better, and quality care that's culturally relevant to their clients, which will, of course, impact on their healthcare outcomes. Quite often, when people do not show up for another appointment for a health checkup or something, it's been documented in the literature that one of the reasons it's simply lack of cultural sensitivity of the staff providing care to clients from cold background. So it's a barrier, lack of cultural sensitivity. That's why it's very important to address this issue at an early stage with our students, with our undergraduate nursing and midwifery students. And in particular, using simulation-based learning is found to enhance cultural awareness, communication, and confidence among nursing and midwifery students by engaging them in realistic scenarios that can develop their practical skills and improve their ability to interact effectively with diverse patients. Simulation is found to enhance understanding of cultural diversity, allowing students to appreciate cultural difference and provide more sensitive care. They learn about specific cultural nuances. Role play in particular, that's one type of simulation, improves verbal and nonverbal cultural communication skills, and this is achieved through realistic interactions, which helps students adapt their communication styles effectively. Students also report after using or practicing their cultural skills through the use of simulation in the form of role plays. They reported feeling more prepared, more confident to engage with culturally diverse patients and exhibiting greater confidence and empathy towards cultural diversity, which of course contribute to their overall professional growth. The most types of simulation mostly used to promote cultural empathy in the literature includes role play, which I just mentioned, where students assume different roles to simulate real life scenarios, which again is found to enhance their understanding of diverse perspectives and improve their communication skills. There is also the standardized patients, where trained actors portray patients with specific cultural backgrounds and health conditions, providing students with realistic, again, interaction opportunities. And there is a virtual simulation where there is interactive computer-based programs that simulate clinical settings, allowing students to practice cultural competency skills in a safe and controlled environment. And there is also case-based learning where students analyze and discuss real-world case studies involving cultural diversity. And this type of simulation fosters critical thinking and problem-solving skills. It's important that educators prepare students for culturally aware practice. Role-play simulation builds cultural awareness and empathy, critical thinking and decision-making, problem-solving skills. All this is done in a safe learning environment where students are allowed to practice as many times as they want through role play. Cultural intelligence is described as an individual's ability to interact and perform meaningfully in a multicultural context, and is found to predict personal adjustment and adaptability, judgment and decision-making, negotiation, effectiveness, trust, and idea sharing among the groups. On the other hand, emotional intelligence enables students to recognize, regulate, and respond to emotions, which is a foundation for empathetic care. So it's all connected. Cultural intelligence equips students to interpret and adapt to diverse cultural norms and patient expectations. Together, emotional intelligence and cultural intelligence support the development of cultural empathy, enhancing communication and trust in cross-cultural clinical settings. Simulation-based learning and reflective practice help students integrate emotional intelligence and cultural intelligence into real-world patient interactions, resulting in promoting cultural empathy and better cultural understanding. Educators play also a role in fostering cultural competence among their students. These are essential for effective educator-student interaction. Lack of sensitivity can harm students' self-efficacy, confidence, and academic performance. Educators must also reflect on their personal bias, prejudice, and discriminatory attitudes, because we're here teaching probably not just in Australia and other cultures or countries. The educator we provide service or education to a diverse students cohorts who come from different cultural backgrounds, and it could be our own attitudes, whether it's bias, discriminatory attitudes, can have a negative impact on how we relate to our students who come from CALD backgrounds, which of course will have a negative impact on their feelings of belonging and their academic performance. So we have to actually be a role model for our students, start by ourselves, demonstrating, our cultural sensitivity, respect to students' diversities, their cultural, religious backgrounds, and, we make it more of a student-centered learning experience. There is a gap in literature addressing the use of simulation to promote cultural awareness and develop cultural competency among university students. In addition, there is a limited research exploring academic stuff, attitudes, knowledge, and skills related to cultural diversity. So it's not just the students, it's not like a blind leading a blind. It's very important also for staff, academics, or faculty staff to become culturally sensitive and aware. In order to reflect this in the way they're teaching, and even to include cultural sensitivity topics in their weekly, not necessarily every week, let's be realistic, but at least for 1 week, allocate a module, a teaching and learning module or lecture or a tutorial material, at least that covers one activity related to cultural diversity, whichever the area or specialty of students, whether they're OT, medicine, whether they're social work, psychology students. It's very important to introduce and promote cultural sensitivity, among our students, because they're the future practitioners, healthcare practitioners, and they're gonna be providing and having to deal with their clients, their patients who come from CALD backgrounds. So it's very important that we instill positive attitudes and regard to people who come from diverse cultural background, because we will influence our students who are the future healthcare professionals. All right. I'm gonna share with you in the following slides a project that I started, or I'm leading with other academics from our school. It started last year, but it's quite a large project. So we were doing it on phases, different, phases. But just a quick background about what interested me, to go ahead and conduct a research study among our students and our academic staff, exploring their understanding of cultural diversity and so on. I was a transcultural mental health CNC, and even my doctorate degree was on CALD. It has a CALD flavor, but with a mental health focus. And I've seen lots of students struggling who come, especially who come from different cultural background, and sometimes they would come and raise concerns of some issues that's impacting on their well-being and obviously their academic performance. So it made me reflect on my own practice, and and start reading about what can we do about it, rather than just going ahead with the classic standard traditional lecture and the tutorial activities that's lacking cultural components in it. So I was invited by scope of practice simulation, like a team, to talk about my project. And this workshop was attended by about 10 faculty staff, including the dean, our dean, and the director of academic program. And I proposed this research study to use simulation, to promote cultural sensitivity. I didn't know what type of simulation, I might be using, and what specific cultural aspect we need to be addressing. And so we had a bit of a brainstorming, like session, and the majority of the faculty staff agreed that using role play scenario, role play, is the best way to promote cultural, like empathy. And again, the issue of being empathetic. We're healthcare professionals. It's been lacking, among, like healthcare professionals. And that's not just what we think, but it's evidence-based, like, as I mentioned, I think earlier in my earlier presentation, that the patients complain and they're becoming more informed now, like they have a stronger voice, and we have patient surveys, satisfaction surveys that they can complete, and we find out that they're not happy with the quality of care that they're receiving due to lack of cultural sensitivity. And the staff, can be rude and not considerate. So the word empathy, or like came up. It's not a concept. I can't even describe what empathy is. It's just being empathetic. It's an essential, skill or human need, really. I think that's the best way to say it, really. It just came like that without thinking about it. Being empathetic, it's a humanistic thing. And when we're talking about we are healthcare professionals, so special nursing, we say it's humanistic, career. It's not a job, it's a career. Being compassionate, providing compassionate care to those who are in need, including people from cult backgrounds. So we agreed that I'm gonna lead this research team, and focusing on the use of simulation in the form of role play to promote empathy, empathetic behavior and practices among nursing and midwifery students. And these are during this workshop, these are some of the ideas, that's been, brainstormed and found to be essential to include it when in the development of these case scenarios. The norms, the beliefs, the values, attitudes, especially those related to help-seeking behavior, perception of illness and health-related issues, the role of religion and spirituality in coping, viewing and management of sickness, how people view and manage someone when he or she gets sick, family dynamics, family interactions, the roles in the family, gender issues, including role in the family, male dominant issues, age issues, again, in some cultural group, in the traditional cultural group, the older you get, the more status you gain. People respect you more because of your age, versus other, in Western cultures, we value youth, the young ones, over the elderly people, which is a bit sad, of course. Again, cultural, some aspects of cultural dimensions, such as power relationships, individualistic versus collectivist societies. Again, in Western cultures, we're more of individualistic societies. We value the person, so it's not unusual to find someone who's sick going by himself or herself to see the doctor or to undergo surgery and so on, versus people who live or who come from collective societies. They come with a whole family surrounding the sick person. So we agreed on these elements to be, included in the development of the scenarios. Being mental health background, that's my specialty. We thought we piloted in one of the subjects I'm coordinating, and we developed a scenario related to cultural diversity for a person with mental health presented to a mental health service, and we developed two scenarios: the good and the bad. The good nurse, trying to interview the person who come from different religion and cultural background with limited English. The scenario is quite long. I didn't want to share it with you, but that's just in a nutshell. And the way the nurse was communicating, it actually acted as a barrier for the patient to open up to her and have a good, like a report and good engagement, with the mental health profession or the nurse, versus a good scenario where there was, the nurse was becoming more appropriate and culturally and religiously sensitive to the patient's needs. We had students and staff working collaboratively in the co-design of these, cultural simulation scenarios. I had a couple of interviews or sessions, I guess, with students who provided feedback on the scenario, and we modified it based on this feedback. It was a good process. It enhanced, the quality teaching or to improve the, the quality of the teaching and promoted student-centered learning. And that's, nowadays at university, we're emphasizing, that, education needs to be student-centered, that we have to involve the students in the co-design of even their assessment, their own assessments, in the content, what they want to be taught. It's quite empowering for the students, and of course, this type of activity, when we did it, we implemented, we piloted in my subject, it increased their engagement in the learning, and during the class interaction, when I was watching them, observing them, not watching, observing them, everyone was engaged in the role play, and they're having turns. One would act like the interviewer, the nurse, and one would be like the patient, and one or two other students, observers, and they rotate among each others. And of course, we had a briefing and the briefing session before and after the simulation activity. It took about just above an hour, which is quite, good. Otherwise, again, from what I've read in the literature about this type of simulation or role play activities, it's not effective if, we haven't given enough or adequate time to do the debrief at the end of the scenario, what was learned and how did they see the activity, how it changed their attitudes and the knowledge, about cultural diversity, what new ideas, what have they learned about the other culture, especially when I'm looking at the diversity in the classroom, the diversity of students, cultural background. It's quite interesting also. I think it made it more, exciting and engaging and rewarding that, they're learning from and from each other's, cultural background due to this diverse group of students, because they come from different cultural background and linguistic backgrounds. Of course, it went through ethics, and yeah, the students, just simply assisted us in the writing up of these scenarios. So the aim of the study that we just, like we got ethics approval last year, but because it's a big project, as I said earlier, we're doing it at different stages. But these are some of the aims: exploring the cross-cultural awareness, skills, and attitudes of students and academic staff. We had different, two groups, one for, and, like survey questions that, specifically addressed academic staff, and others for the students. Examining also the use of assimilation in education and to evaluate the effectiveness of cultural assimilation in enhancing students' cultural awareness and empathy when caring for or providing services to clients from called backgrounds. These are some, it's most of the research questions. What is cultural intelligence level of nursing students? I use the cultural intelligence scale, part of the survey. The survey had 4 components: one demographic data, the standard demographics, one on cultural intelligence, and another one on emotional intelligence, and the third survey was on the scale of empathy. So that's here. Let me go back to mention the other research questions. Is there an association between cultural intelligence, emotional intelligence, and empathy and feeling of compassion in nursing students? What is the effect of cultural assimilation? Education on students' empathy, cultural intelligence, and emotional intelligence. We had the post-education evaluation for the students, and that's what I'll be focusing about in the next a few minutes or so. And this is how far we're reaching with the progress of this project. We collected data from both from students. It's a bit slow with the academic stuff, which is a bit sad. Like I have to keep pushing. Yeah, but we collected adequate number of surveys from the students, and as well as for the post-intervention, post-education, survey, evaluative survey. Is there a relationship between students' cultural competency, emotional intelligence, and empathy? Is there a relationship between certain demographic variables such as cultural background, the years living in Australia, prior education, especially for the pre-registration group involved in this study? Most of them, we have some doctors, some engineers, from different psychology, from different, disciplines. Coming and doing their course with us. Exposure to other cultures, whether they traveled abroad, they're familiar with other cultural, gender, religion, age, and its association or relationship with being empathetic, more empathetic or less, and emotional intelligence as well as cultural cultural intelligence. It's a quantitative descriptive, study. We use surveys. That's here the actual name, the correct names of the surveys we used. The cultural intelligence scales by Dine and All. The should self-report emotional intelligence test. The scale of ethnocultural empathy, the C by Wang and All, and post-intervention or education survey. That's satisfaction with cultural assimilation scale to assess the effectiveness of the cultural assimilation in the form of role play. Initially, we 300 students enrolled in the pre-registration nursing course were invited to participate in the study. 270 students completed the surveys, these 3 surveys, but only 170 students completed the post-education survey. That's the cultural assimilation experience scale. That's we're still at an early stage, but we had some preliminary findings that's related to the debriefing and the facilitation. That's the evaluation. That's what I'm sharing with you now is the outcome of the evaluative survey. The post. Sorry, I've been at work from 6 o'clock in the morning. It's actually now 8 almost 8:30 at night, and I've been teaching and sitting on the desk. My apology if I got sore neck. Sorry. So yeah, the evaluation focused on the briefing and the facilitation. Participant appreciated the structured debrief and felt comfortable asking questions. Feedback during the debrief was seen as constructive and informative. In terms of cultural awareness and empathy, they found that, or many of the students reported increased empathy and awareness of the challenges faced by culturally diverse clients. The realism of the scenarios helped students connect emotionally. In terms of confidence and application to practice, students felt more confident interacting with people from different cultural backgrounds. And several students noted they could apply the lessons learned directly to their future professional practice. So that was a very good point, actually. Relevance and engagement. Most students found the scenarios relevant and reflective of real life situations. And the activity was described as immersive and thought-provoking. Overall, the cultural simulation was engaging, valuable, and held their interest. So that's a very good step, our outcome. It was more, I think, what made it, I straight away got a direct feedback from the students' engagement, active engagement in the classroom, they're chatting, they're laughing a bit. So it's a sign that learning is taking place and they're becoming active learners. So that was quite a good thing to observe. There's some recommendations here, especially for curriculum, to integrate simulation-based learning into nursing and midwifery curriculum. And of course, this can apply to any other health specialty or even any other area. I have contacts with academics from the School of Business and Accounting and Law, and they showed interest in actually adapting the cultural simulation to enhance their cross-cultural communication. If they're working in a bank, a firm, if a lawyer, how to communicate effectively with their clients, whom they're presenting them in the court of law, and so forth. So it has relevance to other and even not non-health related, like disciplines. So that's quite good. I made the change to the ethics. I made some changes requesting approval, of course, to become a university-wide, project, and I got approval for it. So good luck to our team, and we'll see. That's another step. As I said, it's we're doing it at phases. Faculty training, again, we need to provide educators with specialized training in designing and implementing culturally focused simulation, enabling them to effectively facilitate learning experiences for students. It's like we're learning together, especially if the academic or the faculty staff needs a bit of a better understanding of cultural diversity to better provide therapeutic and good communication and interaction and relationship between that's a student teacher, student academic, student faculty staff relationship, which will definitely can make or break, the relationship as well as students' performance and academic performance. And we want to keep our students. Retention, that's actually another good thing, when the students are happy with the quality of not just the education we're providing, it's how. I always emphasize it's about how we communicate, how we teach. It's not about what we're teaching or what we're saying. It's about the how, the process of teaching and learning rather than the content. The content they can read. Even I tell them in the classroom, you can read. Your adult learners, your university students, you have the mental capacity and the cognitive ability to actually read things by yourself on your own time without having to come and sit to what I have to keep talking to you about. and especially we're encouraging self-directed learning and being, following adult learning principles. They're autonomous learners and so forth. So I think it's quite empowering and emancipatory, which are relevant to adult learning, principles. We're not teaching school kids, school, age, students. We're teaching mature students and adults. So we have to stick to these beautiful adult learning principles and apply it in our practice. So it's good to emphasize on the processes of learning. We need to conduct more longitudinal, studies to assess the long-term impact of simulation-based, training on cultural competency among nursing and midwifery, professionals. So maybe need to follow up upon graduation or when they start, their clinical, practice as practitioner, to see actually the impact of the prior learning during university on the long-term retention of and understanding and application and implementation of what they've learned in the classroom setting and transferring this learning into real life, and in their workplace setting. This is when we say, yes, we did a great job. That's thumbs up. So what's the, next? We're planning to develop generic instruction or manual facilitator's guide for various simulation activities, especially if we're doing it, university-wide. So it's good to have something standard, but of course, any faculty staff, whatever their specialty is or school is, they can adapt it and modify it to meet their own specialty needs. Another part of the project, again, that we got ethics approval for, is to carry out an audit on subject outline to map the assessments, the content, the learning outcome, to find out if cultural awareness and cultural competency is included in the weekly, content, in the assessments of students, and if simulation activities, are used or not. What is the structure, the type, and the content of the simulation activity being used? Again, based on students, evaluation, when we had this consultative or when they were helping us in the co-design of these scenarios, they actually recommended, one of the recommendations I'm gonna make to the school, I'll write a report and present it to our school, is that they actually requested that it's a must that cultural simulation, especially in the form of role play, to be embedded in all subjects. So that's quite, also a good feedback, direct feedback from the students who assisted us in the development of these scenarios. That's it. Came to the end. Maybe, hopefully, in the next conference, I would present you with the actual results of the other surveys that we used. That's just a preliminary finding and a summary of the project we're carrying out here in our school at the moment. Any question? Thank you. Thanks for listening.
Yes, thank you so much, Juhani, for your wonderful presentations. If you have any questions to Jihanna Andrews, please unmute and ask her directly to answer your queries, or you can place them in chat box so that she can answer.
May I apologize because I've got a third presentation as well. I think it's too much for the participants here, to listen to a third presentation. Maybe we leave it for our next conference, if that's okay with everyone and with you.
Yes, sure. That's okay. That's fine.
Thank you. Appreciate it. Thank you for allowing me this opportunity to share my work and get also feedback from you guys, especially for the first presentation. I'm actually doing, sorry, I just forgot to mention that I'm doing actually a research project interviewing victims or survivors. I shouldn't say victim, it's a bit negative. Survivors of domestic violence among called communities, and I'm also doing another separate project target looking at the mental health of the LGBTQ plus communities and domestic violence issues as well. So I've got
Yes,
few
yes, that's
to share
great.
with you in our next conference. So wonderful to have you all guys, and thanks for listening to me. Thank you. Thank you
Yeah,
very much.
sure. We look forward to your contribution to our next conference as well.
And have a good night because it's 8:30 p.m. in Australia.
Yes, yes. Thank you so much for your excellent presentations and the content you shared with our audience.
Thank you. Bye, everyone.
May, may I just say thank you so much, Ian, for all the talks, likewise, Omar. And I just want to say that I find your presentation so inspiring, and um, just in case you would like to know how such data sets may look like on this side of the globe, feel free. I would be glad to look into what that happens through the segment of our research and NGO center, because we take a lot of interest in domestic and family violence. And I think you're doing something tremendously well. And thank you again for the presentation.
Thank
Thank
you.
you, Innovine, for the opportunity.
Dr. Abia, thank you. It's good. I don't like if we can exchange, like emails or contacts. The organizing committee or people, the organizers. I'm happy to share my contacts with you, or if you can send me your contacts. I think it's here in the program. I'll find it.
Absolutely.
Keep in touch, and we can do collaborative work. Who knows?
Absolutely.
Thank you. All the best. Thank you.
Yes, thank you so much, Jehane. We hope to see you again at our physical event. Yes, next I would like to invite Gabriel Vaccariu from Bucharest University, Romania, to present his talk on Is Cognitive Neuroscience a Pseudoscience?
Hello. Thanks for your invitation. I would like to have a short presentation about the main topic. Is cognitive neuroscience a pseudoscience? And this is my presentations. Uh, uh, I'm from Bucharest, Romania, somewhere in Europe. What is a science? Because the title is cognitive neuroscience is a science or a pseudoscience. What is a science? Well, a science, it's very difficult to give I'm in philosophy, I work in philosophy, so the definition of science is quite difficult, but I suppose that there are some entities, physical or other kinds of entities, process interactions and certain laws, like in physics, microparticle or planets, biology, life and organism, neuroscience, brain, psychology, mental states and the mind. And in philosophy, we don't have science, it's philosophy. Uh, my presentation will be under the umbrella of the mind-body problem. And the mind-body problem means in the past, dualism from Descartes, dual aspects theory by Spinoza, identity theory in the last, let's say, 70 years, and other approaches. But I want to emphasize that all philosophers and scientists until 2005 have been working under a wrong paradigm, universe world. I call this wrong paradigm, universe or world, uh, unicorn world, because the universe does not exist. Where is the universe? Look, I see these walls, the table, the chairs. No, this is not the universe. It's part of the universe. No, wait a moment, it's a part of the universe. The universe, there is the chair in front of me. There is that stone, but where is the universe? Universe is composed of all these elements, planets and microparticles, but the universe does not exist. Anyway, so I applied this to cognitive neuroscience. My specialty is in cognitive neuroscience and physics, philosophy of physics. Cognitive neuroscience, CNS, under the mind body problem, identity theory, place, and smart. In the last 20 years, many people, specialists, use correlation, like Omar in his presentation about language. It was in the title correlation between mental states and neural states. The question is, but what does mean correlation? I correlate a mental function with speaking, or language with some parts of the brain. But what does mean correlation? Does correlation has an ontological status? And I will show immediately that there is no ontological status correlation. If we work under the identity theory, some certain mental functions are identical with some neural parts of the brain. The correlation is just an epistemological notion. So in fact, in physics, we have no law. There are no laws. There are only entities and interactions. Tell me a law in physics. Oh, gravitational law. There is no gravitational law. There is no gravitational force. It's just space-time curve. As Einstein said, no, it's not space-time, because space-time does not exist. I showed in the past this. Anyway, cognitive neuroscience. In cognitive neuroscience, we relate neuroscience and psychology, but what are the entities of cognitive neuroscience? It's just correlation between mental states and neural states. So we have no entities, we have no laws in cognitive neuroscience. Maybe we can extend the definition of science for cognitive neuroscience. But wait a moment. How do we define correlation? I showed in my books in the past and articles that many problems from cognitive neuroscience are pseudo problems constructed in the wrong framework. So, because there is no ontology in cognitive neuroscience, correlation has no ontology. Cognitive neuroscience is the pseudoscience. There are no law, and in this context, I came in 2002, 2005, 2007 when I published my paper, PhD thesis, with a new framework of thinking, epistemology of different worlds, for cognitive neuroscience, physics, and biology, and philosophy, of course. Universe does not exist. Space-time doesn't exist. Infinite does not exist. God cannot exist. There is a free paper published by me. God cannot exist. Doesn't matter which God. But what there are? There are epistemology from worlds. What does it mean? Epistemology from worlds means there are entities that compose an epistemological world. The epistemological world doesn't mean that it's something in our mind. They really exist, like the table in front of me and the stone and my phone. I put my phone on the table. Yes, the phone interacts with the table. There are macro entities like planets and stones and mountains and rivers. Uh, in front of me is the table and the chair, but in front of me is already an amalgam of microparticle. The table corresponds to an amalgam of microparticles. So what is the relation between microparticle and the table, macro objects? And until me, they said, no, macro objects are the microparticle. So it means that they don't exist, macro particles, macro entities. I said, no, macro particles are in a macro epistemological world. Microparticles are in an epistemological world, micro epistemological world. And we have quantum mechanics with electromagnetic waves and fields, and there is another epistemological world, field electromagnetic. So I applied this to mind-body problem, mind-brain problem, mind-body, and I said that mind is an epistemological world. It is in itself and for itself. It does not exist even for the brain. Mind is immaterial and body is material in the macro epistemology. Where is the table and what is the stone and so on. And there is no relation between mind and brain, because one does not exist for the other. Mind is in itself and for itself. It doesn't mean that mind does not exist. It exists, but it does not exist in the world of the brain and the body. So that we have only correspondence, but the correspondence is related with correlations. Correspondence has no ontology. So my phone interacts with the table in the macro world. Yes, a microparticle from the light interact with the microparticle of the table. The problem is that the microparticle does not exist for the macro, and the macro does not exist for the micro, but both exist in observational worlds. I apply this to Einstein versus quantum mechanics, planets and the microparticles. All interpretation from quantum mechanics have been wrong. I replace with a new interpretation of quantum mechanics. I don't talk about this. And I apply to cognitive neuroscience, physics and philosophy about ontology, all this quantum gravity, dark matter and dark energy. You can find some papers free and many of my books free on internet in English. So in cognitive neuroscience, we have very important problems in cognitive neuroscience topics like localizations, binding problem, perception versus cognition, multisensory integration. Somebody talk about very shortly about mind reading and so on and so forth. We use fMRI, PET, EEG, MEG, and others to grasp parts of the brain. The problem is that I take an example. I see in front of me a green table, a green tree. Where is this visual perceptual image? It is in the brain. Wait a moment. I don't have green cells in the brain. How do I see the green of the trees, of the leaves? In the brain, there are some chemical and electrical activities of the neurons and so on and so forth. Wait a minute. When I see the green tree in front of me, I don't see some neurons activated. I see where is the green? Green is not on the leaves because it's just a reflection of light with some frequency and wavelength. So where is the green? It's not on the tree. It's not in my brain. But I see the green. So I have a perceptual image of a green tree. Where is that perceptual image? It is in my mind. But my mind does not exist for the brain does not exist for the mind. So we have in cognitive science, computations, connections, dynamic system approach, different interpretation of the brain and mind and so on. I don't talk about too much about this. It's just that each mental state is part of the self or the mind and correspond to more or less parts of the brain activated. But in fact, when I see that green when I see that tree in front of me and it appears in my mind as being a green tree, in fact, my entire brain and the body is involved in perceiving that the mind and brain correspond to not mind, brain and the body correspond to the mind. So my entire brain is activated and the activation of my brain correspond to my mind, and I have a perceptual image of the tree. For instance, I see a green card in motion when and has a noise. This is multisensory integration. Wait a moment, I have the noise from the ears and the green image from the eye. So somewhere in the brain has to be an integration of this multisensory information. And I showed that there is no other part of the brain which localize this integration. In fact, there is no integrations because the mind corresponds to the brain, but mind does not exist. So that integration from the ears and the eyes, there is no place. There is no mechanism for such integration. It's in fact the mind which corresponds to the brain in which appeared the unification of information from the which correspond to the ear and to the mind. So any visual perceptual state of a green table is the same, even the neurons and brain change the reactivity. Look, I see this the table in front of me. Nothing changed in my image, but in the brain, when I see 10 seconds this table, in the brain, there are a lot of changes. Even if, even in the most activated neural patterns, some neurons change their activation. So why nothing change in my visual perception in that moment? Because the visual perception does not belong to the brain, it belongs to the mind. So self or the mind or the life are the same thing. We have a representation and computation. These are very strong topics in cognitive neuroscience and philosophy of mind. I don't talk about this. We have different knowledge, implicit, explicit, declarative, procedural, consciousness, unconscious, serial, and so on and so forth. And we have different kind of representation, propositional representation or pictorial visual representation like images and map and so on and so forth. Mind reading, fMRI, EEG is a very approximate process. There are just correlation between mental images and neural parts of the brain that are activated, most activated. But we don't have only neural activation. We have electromagnetic waves. We have a lot of chemical reactions in the brain. So when you use fMRI, we grasp just some aspects of the brain that is activated for central mental function, but in corresponding. But in fact, there are a lot of processes in the brain that are activated, not only electroencephalogram, not only electromagnetic field, but are chemical reactions, and neurons send electrical impulse to the other neurons, but we have other activation in the brain. So using fMRI or EEG or other pattern, we just grasp parts of the brain activity. Other notion in cognitive science, levels, emergence, modularity, perception. I in my books I wrote about all this. Are a lot of different. So my very short presentation is just about this. Mind is an epistemological world which exists in itself and for itself, and brain body is an entity in the macro epistemological world. Where is the table? Where is the car? Where is the planet? And what is the relation between mind and brain? Doesn't exist, because mind does not exist for the brain does not exist for the mind. So we have special cases of brain, body, and personal identity, like twin sister Krista and Tatiana shared a single brain. Twin sister Abby and Bretty, two minds, two brain, but a part of the body is common. Kim Peek with Rainman seventh syndrome. And I have a twin brother. I have a twin brother. I wrote 23 books. My brother wrote only 2 books. We were born in the same family, in the same environment. So there are no genetic nations or individuals, because the difference between me and my brother is quite huge regarding the publication of the books, not about intelligence, because we are quite similar. But I want in the end to talk about a particular case, brain damage myself. In Oxford, I had an accident. I fall from this second level of a building because the key didn't work and I couldn't open the door. I was with a scholarship there, and I climbed through on the wall, because I climbed the mountains, and I said, I can't climb the wall, and I fall from the second level. I was in coma 10 days, but it was an artificial coma. Anyway, after I recovered, and after I recovered, after 8 years, I started to have epilepsy, but it was after 8 years. It was quite strange for specialists in epilepsy. Anyway, I believe that in fact, as somebody said before me, each person has her brain. In fact, are very different. The identity, personal identity, is very different, because the brain is very different between us, because our experience of is very different in our entire life. So our mind, each has his mind. My mind does not exist for your mind. Your mind does not exist for my mind. But each has her own mind. So this is the end of my presentation. I have published 24 books on different topics, and I applied my epistemological words to many topics: non-locality in quantum mechanics, and the Einstein versus quantum mechanics, and all topics from cognitive neuroscience, mind, brain, and others. Is cognitive neuroscience a pseudo? Yes. We don't have any entities in cognitive neuroscience. We don't have any laws. Anyway, the laws does not exist even in physics. But we don't have any ontology in cognitive neuroscience. We have ontology in neuroscience. Yes, there is the brain, neurons, and so on and so forth. We have some entities in psychology, like mental states and mental representation and emotions, and so yes, we have. But when we try to put together mind and brain, this is cognitive neuroscience. We try to relate two entities which does not exist: the brain, body, and the mind does not exist one for the other. So how we can create a science which relates something which does not exist one for the other? Brain, body does not exist for the mind does not exist for the brain. So how to create a cognitive neuroscience for this. So it is a pseudoscience, and all the problems in cognitive neuroscience are pseudo problems created since the 1970s until our days. So I end with Albert Einstein, a clever person solve a problem, a white person avoid it. So it's better to avoid long, not solved problem. It's better to find that that problem is a pseudo problem and not to try to solve it. So this is my presentation. Thank you very much for uh, your presence. Thank you.
Yes, thank you so much, Gabriel, for your excellent presentation.
So if there are any questions,
Yes, if you have any questions to Gabriel, please post them in chat box, or you can unmute and ask him directly to answer your queries.
okay, thank you very much for your presence.
Yeah, thank you so much, you too. Yes, moving forward to the next presentation, I would like to invite Paula Abola from the University of Jamestown from United States of America to present her talk on technological advancements in the detection and quantification of Parkinsonian tremor, a scoping review. Due to, due to her unavailability of Paula Abola, she has shared the recorded presentation.
I'm sorry, I can't hear the presenter. I can hear somebody in their kitchen and some traffic.
I think the host can mute the person if you know who from where this noise is coming from. It sounds like Egypt, my country. It's like that. The noises.
Yeah.
It's dinner time. I can hear.
Yeah, I can't hear anything as well.
I'm sharing is caring.
Yes,
Huh?
sorry for
Sorry,
the
breakfast,
interruption.
dinner, lunch, whatever it is. It's funny.
Yeah, and I think the presenter is muted.
Yeah, I can't hear anything either.
Yes, sorry for the interruption. We are sharing the video presentation once again.
I still can't hear her.
Hello everyone, my name is Paula Abel, and I'm honored to present the scoping review conducted with my colleague. Today I will walk you through a recent technological advancements in detecting and quantifying Parkinsonian tremor, and how these tools may support individualized rehabilitation for people living with Parkinson's disease. Tremor is one of the hallmark motor symptoms of Parkinson's disease. More than 70% of individuals with Parkinson's disease experience tremor, and many report it as the most bothersome symptom. It affects daily functions like eating, writing, or dressing, and often causes social withdrawal and anxiety. Despite the prominence of tremor, there is no universal tool to objectively quantify it in daily life. That's where emerging technologies like sensors and wearables may offer a solution. This review had 3 objectives. First, to identify what technologies are currently available for detecting and quantifying Parkinsonian tremors. Second, to evaluate how these technologies are used in practice, especially for rehabilitation and monitoring. And finally, to identify where evidence is lacking to help guide future research. We conducted a scoping review following the PRISMA 2020 systematic review guidelines. We searched 3 databases, PubMed, Cochrane, and EBSCO. Out of 272 initial articles, only 9 met all inclusion criteria. We included studies involving individuals with Parkinson's disease experiencing tremor, using technologies such as accelerometers or sensors for detection or quantification of tremor. The 9 included studies covered 248 individuals diagnosed with Parkinson's disease experiencing tremors. Some studies compared individuals with Parkinson's disease to healthy controls, while others focused solely on patients with Parkinson's disease. Technologies ranged from wrist-worn accelerometers and smartwatches to even textile gloves and mobile phone apps. We reviewed different types of technologies. The most common were accelerometers, either in wearable form or built into smartphones. Other studies used force or inertial sensors, or even specialized gloves embedded with multimodal sensors. A few studies combined smartwatches with smartphone apps for home-based monitoring. Accelerometers were the most widely used and studied technology. They detect tremor by capturing movement during daily tasks, either in a lab or even at a home setting. Studies reported high accuracy of up to 98% in detecting tremor episodes. Some devices even capture changes between medication states, which is essential for tracking symptom fluctuations. Force and inertial sensors add another layer of quantification. For example, some studies used pinch force sensors to track tremor severity under cognitive load. Other studies used the so-called power glove to assess multiple hand and wrist motions. These tools offer detailed insights into tremor patterns that can be used to fine-tune both medical and rehabilitation interventions for people living with Parkinson's disease. A few studies looked at changes in tremor before and after medication or exercise. In one trial, eccentric hand training led to a 63% reduction in resting tremor. In another, a smartphone system showed significantly higher tremor scores when participants were off their medication compared to when they were on it. And these findings point to the clinical relevance of quantifying tremor across different treatment conditions. Currently, the Unified Parkinson's Disease Rating Scale is the standard for assessing motor symptoms, but it's subjective and limited to clinic visits. In contrast, wearable technology provides continuous real-time and objective data. This allows clinicians to monitor symptoms remotely, adapt to treatment plans, and respond faster to symptom changes. These technologies aren't just diagnostic. They can also be integrated into rehabilitation. They help clinicians design personalized interventions based on real-time data and monitor how patients respond. They're also useful in remote settings or when access to in-person visits is limited. Future directions include collecting more real-world data, especially outside of structured clinical settings. And there's also great potential in combining these devices with other technologies like virtual reality or neuromuscular electrical stimulation. And beyond tremor, similar tools may help quantify bradykinesia, gait issues, and even dysphagia. In conclusion, technology is rapidly transforming how we assess and manage Parkinsonian tremor. These tools support earlier detection, enable personalized rehabilitation, and empower clinicians and patients to track progress over time. Ultimately, they hold promise for improving quality of life in people living with Parkinson's disease. Thank you for joining my presentation. Feel free to email me at paula.arbola@uj.edu if you have any questions, or of course connect with me on LinkedIn by scanning the QR code on the slide. And thank you so much again.
Well, that was a presentation from Paula Arbola. Yes, moving forward to the next video recorded presentation from Kathleen Lehman from the Chicago School of Professional Psychology, United States of America, to present the talk on neurodynamics and clinical implications of heart-brain coherence and non-sleep deep rest. And again, due to her unavailability, we would like to play the recorded presentation of Kathleen.
Welcome, everyone, and thank you for the opportunity to share this work with you today. The focus of this presentation is on a set of physiological states that hold surprising power for healing human systems and transformation. What we call heart-brain coherence and non-sleep deep rest, or NSDR. These aren't just wellness buzzwords, they are measurable neurophysiological phenomena that can be intentionally cultivated. And they may offer one of the most accessible, low-barrier pathways we have for regulating the nervous system, improving health outcomes, and restoring resilience, not just at the individual level, but across our social and ecological systems. Today, I'll be offering a deep dive into the underlying mechanisms, the emerging clinical applications, and the broader implications for how we approach mental health, systems care, and the future of medicine. Let's begin with the context. Our world is in a state of chronic physiological arousal. We are witnessing a surge in stress-related disorders like anxiety, depression, burnout, autoimmune conditions, and trauma-related dysregulation, which are all reaching unprecedented levels. But what's often missed in the public health framing is that this is not simply a crisis of mental health. It's a systems-level breakdown of regenerative homeostasis. The human nervous system is exquisitely sensitive to threat. Not just physical threat, but social, psychological, and environmental disruption. And today, it's under siege from multiple directions. Global uncertainty, ecological degradation, economic instability, and relational fragmentation. This dysregulation shows up in the body, in disrupted sleep cycles, hypervigilant immune responses, erratic cardiovascular rhythms. It shows up in our collective behavior too, in polarization, attention fragmentation, and burnout in our healthcare systems. So the question is, how do we intervene at a level deep enough to restore balance, coherence, and trust in the body, in our relationships, and in our shared systems? To explore these questions, we need to understand two related phenomena: heart-brain coherence and non-sleep deep rest. Heart-brain coherence refers to a state in which the rhythmic activity of the heart, measured through HRV, entrains with breath and blood pressure in a synchronized, sine-like wave pattern. When this happens, it improves communication between the heart and the brain via the vagus nerve and baroreceptors. Coherence is not simply a relaxation state. It's a state of optimized self-regulation that can be intentionally trained through breathing techniques and biofeedback. NSDR, or non-sleep deep rest, refers to a class of neurophysiological states characterized by parasympathetic dominance, low cognitive load, and maintained awareness. This includes practices like yoga nidra, guided body scans, and flotation rest. What's fascinating is that these states mirror some of the same brainwave and autonomic patterns we see in coherence. Increased theta and delta activity, reduced beta, and high vagal tone. When viewed together, these two practices don't just calm the body, they restore its capacity to adapt, connect, and heal. And this has wide-ranging implications for both personal recovery and population health. So what's actually happening under the hood during these states? From a neurodynamic perspective, coherence is facilitated through vagal afferent signaling, information traveling from the heart to the brain. These signals reach the brainstem and are relayed to the insula, which governs interoception, and to cortical areas like the prefrontal cortex and anterior cingulate cortex, which govern emotion regulation, attention, and executive function. EEG and neuroimaging studies show that coherence is associated with a shift towards theta-gamma coupling, decreased high beta, which correlates with stress and vigilance, and increased connectivity in the default mode network, but with less rumination and more present-centered awareness. In simple terms, when we are in a coherent state, the entire system, cardiac, respiratory, and neural, is operating in rhythm. That internal rhythm, or coherence, appears to promote not just resilience, but adaptability. And this is a key point. Coherence is not rigidity. It is flexible alignment in the face of change. Now let's look more closely at NSDR. These states, in which we are deeply physiologically relaxed, but not unconscious. Research shows that during NSDR, the brain shifts into slow wave states, theta and delta, while high wave or high frequency beta activity declines. This is paired with increased heart rate variability and vagal tone, reduced cortisol, and deactivation of the amygdala, which is central to the fear response. In flotation rest, sensory input is minimized, allowing the brain to enter a hypnagogic state. In yoga nidra, guided awareness helps decouple thought from reactivity. Both produce measurable changes in brain and heart dynamics within minutes. What makes NSDR so powerful is that it allows for integration, a process often overlooked in our overstimulated culture. Rather than forcing insight or change, the nervous system is gently supported into a condition where it can recognize or reorganize on its own. From a systems view, this is restoration of internal ecology. To bridge these two practices conceptually, we turn to polyvagal theory, developed by Stephen Porges. This framework explains how the vagus nerve mediates our transitions between threat, mobilization, and safety. Of particular interest here is the ventral vagal branch, which supports social engagement, trust, and pro-social behavior. Both coherence and NSDR activate the ventral vagal pathway, signaling that it is safe to relax, digest, feel, and connect. Importantly, this is not just an internal state. It is the physiological foundation for relational coherence. Without it, we
we cannot sustain empathy, collaboration, or even collective resilience. In this way, these practices move us beyond the individual regulation. They are also blueprints for how institutions, communities, and societies might begin to regulate with one another again. In clinical settings, coherence and NSDR are proving effective in reducing symptoms of anxiety, depression, and post-traumatic stress. HRV biofeedback has been shown to improve emotion regulation, working memory, and attentional control. NSDR protocols, especially float therapy, have been associated with reduced rumination, increased self-compassion, and improved affective recovery. This is particularly valuable for trauma-affected populations. Traditional cognitive interventions often bypass the body, but trauma is fundamentally physiological. These methods allow clients to feel safe again in their bodies, a prerequisite for any sustainable psychological healing. These are not just treatments, they are capacity builders. They increase our baseline resilience and reduce allostatic load, the cumulative burden of stress on our systems. Let's move into another area, inflammation and pain. What we're learning is that the nervous and immune systems are not separate. They are deeply interwoven, with the vagus nerve acting as a communication superhighway between the two. Through what's called the cholinergic anti-inflammatory pathway, vagal tone plays a major role in downregulating pro-inflammatory cytokines like the IL-6 and TNF-alpha and CRP. These inflammatory markers are elevated in chronic pain conditions, autoimmune disorders, and even mood disorders. Both coherence and NSDR have been shown to reduce systemic inflammation and offer non-invasive adjunctive care for people with fibromyalgia, irritable bowel syndrome, long COVID, and neuroinflammatory conditions. Viewed through an ecological lens, inflammation is the body's call for repair. Coherence doesn't suppress that signal. It helps fulfill it, guiding the system back towards balance and integration. One of the first areas where many people feel the benefit of these practices is in sleep and fatigue regulation. NSDR states mimic aspects of slow wave sleep while preserving awareness. This has been shown to increase sleep efficiency, shorten sleep latency, and improve next day energy levels. When HRV coherence practices are done before sleep, they signal the parasympathetic system to take over, allowing the body to wind down naturally. This is especially important for individuals dealing with insomnia, trauma-related hyperarousal, or chronic burnout, where sleep is often compromised even when exhaustion is high. In this context, NSDR is not a substitute for sleep, but it amplifies the restorative power of rest, making it an invaluable tool for frontline workers, caregivers, high-performance populations, and anyone living in chronically activated states. If we think of burnout not as a personal failure, but as a systems failure of recovery, NSDR becomes an ethical intervention, not just a wellness tool. Cardiovascular and metabolic diseases are now among the top global killers, and they are deeply tied to autonomic imbalance. When the sympathetic nervous system dominates, we see elevated blood pressure, increased insulin resistance, dysregulated glucose metabolism, and impaired cardiovascular resilience. HRV coherence training can improve baroreflex sensitivity, helping the body more efficiently regulate blood pressure fluctuations, while NSDR supports insulin sensitivity and reduces systemic stress hormones that impact metabolic function. Imagine if instead of waiting until people are diagnosed with hypertension or diabetes, we integrated coherence protocols into primary care, schools, and workplace wellness as part of a public health strategy to build systems-wide resilience. In this way, coherence becomes more than personal. It becomes preventative medicine for populations. We also need to consider coherence and NSDR in the context of developmental and intergenerational trauma. The autonomic nervous system is shaped during early attachment experiences. If a child grows up in an environment of threat, neglect, or unpredictability, their baseline vagal tone and neuroception of safety may be impaired. These physiological patterns can be passed down epigenetically, not just through behavior, but through cellular memory. NSDR and coherence help to repattern these responses, building new pathways for safety, resilience, and co-regulation. There is enormous potential here for interventions in schools, for prenatal care and postpartum recovery, for programs that support intergenerational healing in communities affected by systemic violence, migration, or historical trauma. We are just beginning to understand how restoring coherence in one generation might ripple forward into others. So what lies ahead? The future of coherence research is vibrant and multidisciplinary. We need more large-scale, longitudinal studies that measure how HRV and EEG coherence correlate with psychological outcomes, inflammatory markers, and cognitive flexibility. But we also need innovation. How might coherence be integrated with psychedelic-assisted therapies, helping guide integration and reduce re-traumatization? What role might wearables, AI, and biometric feedback platforms play in democratizing access to NSDR protocols? How can we design interventions that are culturally adaptive and trauma-informed, ensuring equity across language, race, and ability? This is where systems thinking is essential. We cannot scale coherence through individual behavior change alone. It must be woven into the structures of how we do education, healthcare, organizational leadership, and social care. So let's bring it all together. Heart-brain coherence and NSDR are not fringe. They are evidence-based, neurophysiologically grounded practices that support the health of the nervous system. They work by restoring internal rhythm, increasing parasympathetic tone, and enhancing emotional and physiological flexibility. But more importantly, they offer a systems intervention, a way to begin healing not just the individual, but the relationships, communities, and environments we inhabit. Coherence is not about perfection or peak performance. It's about finding rhythm in the midst of chaos And from there, allowing the system, whether human or planetary, to reorganize towards healing. Here are a selection of references used throughout today's presentation. I want to thank you all so much for your presence, your attention, and your care. This is a conversation that touches not just the nervous system, but the soul of how we think about healing, justice, and human potential. I welcome your questions, collaborations, and reflections as we continue building coherence together. Thank you.
Well, that was a wonderful and informative presentation from Kathleen. Yes, last one for today's sessions. Next, I would like to invite Anusha Naserdahari from University of Tehran, Iran, to present her talk on visual out preferences in schizophrenia.
Hi everyone, thank you for joining my virtual poster session, and thank you to the campus organizers for having me. My name is Anissa, and today I will be discussing our latest book on the topic of art perception in schizophrenia. So the question that we wanted to study was that are there any differences in the way that people with schizophrenia experience and appreciate visual art? Now, there have been a lot of studies undertaken in schizophrenia and creativity. Some suggest that the link follows an inverted pattern, meaning that mild expression of schizophrenic traits are linked to higher creativity, and also particular relatives of people with schizophrenia uh, who possess subclinical traits usually take up more creative jobs. And it's intuitively more understandable to a brain that finds patterns in essentially everything is potentially more creative. But most of the studies on this topic so far have been concerned with the production of art rather than experiencing it. These studies show that artists with schizophrenia have a tendency towards using specific visual elements. For example, these are subject matters, colors in higher contrast, or filling the page with repetitive motifs, and then emphasize on gross structures and bold lines within their art. We know that it's not merely an aesthetic design choice either, because looking at artworks from Louis Spain, for example, who drew cartoon cats, we can see how his art changed and evolved into more symbolic and weird as he progressed. These changes or excessive use of the specific visual elements may be related to alterations in visual processing. For example, things like reduced contrast sensitivity, impaired visual integration, or a change in visual motion perception, all of which are very common in schizophrenia. So one theory is that artists with schizophrenia use these elements, for example, use higher contrast to make their paintings more visible to themselves. But still, we don't completely understand the differences in perception of visual elements and visual compositions in schizophrenia. And that's her second. We wanted to see whether art naive people with this condition show a different appreciation or understanding of visual art. We also wanted to see the effect of mood on aesthetic appreciation and how it changes after engagement with art. So for our experiment, we used the computerized lighting rating test in combination with Panos mood questionnaires before and after the main task. The task design was similar to the one used by Ligoya and colleagues in their 2021 paper. You can check out in the method section. So 150 paintings in 5 different art styles were pulled at random from Wiki Art, everything from each. We wanted impressionism because of its use of vibrant colors and vivid subjects, expressionism for its unconventional subjects, for its focus on conveying strong emotions through exaggerated, distorted forms, and intense colors, very similar to artwork we see from artists with schizophrenia. And cubism because of its structured geometrical compositions and muted colors, abstract art as non-representative art, and color with art to see the effect of color. It's just plain color. And we also wanted to make sure that these paintings are not well-known works of art, so familiarity is an important field in aesthetic appreciation. So the project went through an initial trial phase where 52 art naive participants rated them based on familiarity. We then had 70 hospitalized patients diagnosed with schizophrenia and 31 healthy controls participate. Before and after the main task, as I said, they were given the Panos questionnaire to indicate their art mood. For the main task, they had to rate on a Likert scale how pleasant they found each painting. And the order in which the paintings were presented to them was random to account for the effects of previously seen paintings on their judgment. And of course, they were briefed beforehand that there was no right or wrong answer. We found that the more positive mood before the task was associated with more positive rating for artworks in both groups. We also saw a decrease in negative mood for everyone after engaging with artworks. And about aesthetic evaluations, we had some interesting differences between groups, but I would like to focus on expressionism for this presentation. It had the largest effect size. If we look at the heat maps on the poster, these are a representation of similarity matrices. They show how similar participants within each group rated the paintings, and they have been sorted by style. So the blocks along the diagonal show agreement for paintings within the same style. And the color scale can be understood as the warmer the color, the stronger the agreement in ratings. Now, if you would please focus on two diagonal blocks, Impressionism at the bottom right and Expressionism, which is the block just before it. We can see the highest absolute agreement for both groups was for Impressionist paintings. We can see it, the block for Impressionism is mostly intensely red. Closely after is the block for Expressionism, as I said, the second from the bottom on the right. We can see this block is also intensely red, but only in the schizophrenia group, not in the control group. So what this means is that while consensus for Impressionism was very high, which is expected because Impressionist art is very vibrant, it's very colorful, it's easy to digest. The group difference was more pronounced for Expressionism. Participants with schizophrenia had a more homogeneous, very positive rating for Expressionist art. And in the bar chart at the bottom of the poster, we can see this effect closer. We can see the liking between probability distributions for Expressionism. So it's what it says is how likely it was for each group to choose a certain rating for expressionist artwork. We can see that the majority of responses to expressionist art in schizophrenia group was highly pleasant. In fact, it was the second highest rated art style overall after impressionism. And our control group had a completely different reaction. They had a much more negative evaluation of expressionist art. Most found it very unpleasant. So what is the reasoning for this? As I mentioned before, there are visual processing differences in schizophrenia. There is reduced contrast sensitivity, so maybe people with schizophrenia find the high contrast, intensely colored expressions easier to perceive and therefore more pleasant. It's in line with perceptual fluency. Or maybe it's not the formal elements and the subject matter instead. Maybe it's the content of sorrow or grief and heartache that resonates with me. And another thing that I'd like to talk about is anhedonia. It's a very common negative symptom in schizophrenia that refers to loss of pleasure. So when we looked at the hedonic ratings of schizophrenia patients, we didn't see this symptom influencing the enjoyment of art pieces or their aesthetic motivations, their mood, and their engagement with art. And this aligns with previous research that the ability to feel pleasure in the moment is preserved in schizophrenia, and the impairment seems to be translating that feeling to long-term motivation. It also aligns with theories of aesthetics. The pleasure we get from art is a different quality of emotion. It's liking without needing or wanting. So when the motivation to acquire something is out of the equation, their emotional responses to art appear to be intact. And this all is part of the reason why I believe there is a strong need for future research into the neural basis of aesthetic judgments, especially in neurodivergent groups, because art doesn't just tell us about the mind that creates it, it also tells us about the mind that perceives it. And schizophrenia is a very debilitating, severely stigmatized illness. So art offers a way of connection, a way to move past seeing only deficits and instead appreciate differences in vision and lived experience. And through that, I hope that we will be able to make the world a more welcoming place for neurodiverse individuals. Thank you for listening.
Yes, that's all for today. As we wrap up, today's session for the 16th World Conference on Midwifery Nursing, Neuroscience, and Brain Health, I want to thank each of you for your active participation and thoughtful questions and the energy you have brought even from behind screens. It's been a day filled with valuable insights and meaningful exchanges, and shared the commitment to advancing the care and science. That was fantastic, and we hope to found the experience both enjoyable and enriching. We look forward to welcoming you to more of our sessions ahead, and of course for the next editions of our conferences. Until then, take care, and thank you once again for your valuable participation and being part of this vibrant community. Once again, thank you each and every member from Innovant Scientific Meetings. Thank you. Have a good day. Bye bye.